Good morning. Welcome to Gairdner Science Week 2022 and our Laureate lectures. I'm Summer Wedlock, and I'm the Executive Vice President of the Gairdner Foundation. And it's my pleasure to welcome you all in person today, what an incredible crowd, and to those who are joining us virtually from around the world. Before we begin, I'd like to take a moment to acknowledge that Mars is located in Toronto on the traditional territory of many nations, including the Mississaugas of the Credit, the Anishinaabeg, the Chippewa, the Haudenosaunee, and the Wendat peoples, and is now home to many diverse First Nations, Inuit, and Métis people. The territory is subject of the Dish with One Spoon Wampum Belt Covenant, an agreement to peacefully share and care for the Great Lakes region. This event is part of Gairdner Science Week, an annual week-long celebration of our Gairdner Award Laureates and the impact of their research on human health. I'd like to thank our longtime partners, the Government of Canada through CIHR, the Government of Quebec, and the Government of Alberta. And a special thank you to all of our Gairdner Science Week sponsors who have made this year's events possible. Our platinum sponsors are the University Health Network, RBC Future Launch, Crumble Foundation, and TELUS Health. Our gold sponsors, as you'll see on the slide, are York University, Terry Fox Research Institute, Queen's University, London Drugs, Roche, CAMH, McMaster University, McGill University, Sick Kids Research Institute, the University of Pennsylvania, University of Toronto, Moderna, and Guardant Health. And thank you to all of our sponsors. Without you, this important week of networking, engagement, and education would not be possible. Please join me in a round of applause for our sponsors. I am truly thrilled to welcome this year's 2022 Canada Gairdner Award Laureates to Toronto. As part of Gairdner's mission to inspire the next generation of researchers, we have sent our 2022 laureates and those also from 2021 and 2020 across the country to share their research with universities, faculty, trainees, and high school students. This is always a special part of our programming, and the inspiration of our laureates is much appreciated by all. Today, we'll hear lectures from our 7 2022 laureates about their award-winning research. We will also hear short talks from Canadian early career investigators who were selected by our laureates to present the related research and meet their scientific heroes. Let's begin with our first speaker, Dr. Catalin Carrico. She is one of this year's Canada Gairdner International awardees for her pioneering work developing nucleoside-modified mRNA drug delivery, which is the foundational technology for the highly effective COVID-19 mRNA vaccines that we have all benefited from and have made this day possible. Dr. Carrico is Senior Vice President, RNA Protein Replacement Therapies at BioNTech, Professor at the University of Szeged, and Adjunct Professor at the University of Pennsylvania's Perelman School of Medicine. Please join me in welcoming Dr. Carrico to the stage.
Good morning. I am very happy to be here, very excited to talk about development of mRNA for therapy. I mostly talk about that 60 years which it took to develop RNA for an FDA-approved product after discovering RNA in 1961, 61 years ago. During this 60 years, a lot of things happened. There are some major things, but most of the time it's just the incremental improvement of mRNA, the delivery, and so on. So it started in 1961 when 2 papers in Nature described the label intermediate, which is carrying information from the DNA to the protein synthesis place, to the ribosome. And with this one, we know that the information flow in every cell in our body, as well as all free living organism, is from DNA to RNA and to protein. So Of course, scientists, 1961, they couldn't synthesize RNA, so at that time, we wouldn't be able to make a vaccine, but scientists start to isolate RNA and start to analyze them. First, they looked at different structural elements. In 1972, they discovered that the RNA has a poly-A tail. In 1975, they discovered there is a cap structure. This was a very interesting product there. It's never seen before. There were modified nucleoside in it. And actually, this was the heydays when everybody started to investigating modified nucleosides. And so the SCAP is the Cap1 element, which if you get the vaccine from Pfizer-BioNTech, was incorporated during synthesis. But in this year, in 1975, several papers describing what is the importance of this cap, and one of the things was that this was required for translation. And by the end of the year, Bernard Moss and colleagues also discovered that there are viral enzymes which can put this cap on the mRNA, and if you get the Moderna vaccine, that was actually using these enzymes to cap the RNA. Other scientists further analyzed and further tested out what all of them was isolated RNA. So they could isolate RNA from reticulocytes, and these were globin RNA. And they translated it in lysates, cell lysates. They injected in frog oocyte. Those are large cells. It was easy, and they looked at for translation of these isolated RNA. And in 1978, two papers described that RNA, when it was wrapped in a liposome, then it can diffuse to primary cells and then generated and translated the liposome-encoded RNA. So this was the first time, really, when it was realized that maybe RNA could be used for some kind of therapy. And liposome was introduced by Branham in London, and a colleague in Dimitriadis in London, he delivered RNA to the cell. Prior to that, other scientists delivered with liposome plasmid DNA. And that was the paper we were reading in Hungary when I was undergraduate student and joined the lipid lab. And with my colleague, we isolated phospholipid, and we generated liposome, and we put in plasmid into it and delivered to the cell. So it is important for the students, start with something, because the rest of my life I was remember this experiment and was thinking about it. So it was not RNA, it was DNA, but delivering nucleic acid inside the cell by liposome was a very interesting thing to do. By chance, next step, to make my graduate studies, my PhD thesis, I went to the RNA lab. It happened that my supervisor, who was an organic chemist, actually synthesized cap analogs and sent it to Aaron Shotkin and colleagues in New Jersey, because if you work with something, you need reference material. My role in the lab was to synthesize RNA. Those were short RNA trimers, and we made enzymatically, we made chemically, and this small RNA had antiviral effect. It was just discovered the prior year, 1977, Ian Kerr in London. And I worked together with János Ludwig. He was a graduate student. And again, telling you, students, who's sitting next to you one day will be, or rest of your life is your advisor, because every time I turn to János for advice on organic chemistry. So this RNA, this small RNA, actually naturally also produced in your body. But of course, our idea was to generate an antiviral compound. And because this molecule, when it is generated, can activate an RNase L latent enzyme, and that was believed that this is cutting up all of the single-stranded viral RNA, and this is how it expresses antiviral effect. It's not turned out everything like that, but we still had a problem delivering this small molecule and ending the funding for that. So that's how I get into the United States. And I still worked on this molecule. And we tried to develop it for antiviral effect because in 1985, 86, when I arrived here, the major problem was the HIV. But eventually, we had to use a different kind of RNA, not this small molecule, but long double-stranded RNA, which had some mismatch in it, and this was less toxic than the poly-IC used prior to that, and we treated HIV patients. So, as you can hear here, that all in my life, I just work with RNA, you know, whether I try to degrade with an enzyme or whether induce some interferon with double-stranded RNA, label the RNA, and so on. So I was very interested always in RNA. While I was working on these projects, scientists at Harvard University figured out how you can generate RNA from using a phage RNA polymerase. I don't have time to tell for everything. There is a lot of scientists worked on it. For example, making RNA in vitro in a tube. Scientists tried bacterial polymerases, but turned out that this phage RNA polymerase has very high fidelity, very effective, and you can generate RNA by adding a template, adding the 4 basic nucleotides, and the enzyme. In that case, they also added the capping enzyme, which Bernard Moss discovered, just I showed you. So they generated this RNA, which code for interferon, and scientists there injected to frog oocyte, cultured them, and they found that the medium had antiviral effect. So this was the first time it was demonstrated that messenger RNA made by design, injected into cells, can code for the generate functional protein. A lot of people get excited. And anyway, in the second part of the 80s, most of you were not born yet, but that was a very exciting time because the PCR was introduced. When the T7 and SP6 RNA polymerase was reported, the same year you already could purchase it. So scientists were excited because now that they can use the RNA for therapy. So here I am showing you all of the paper which was published in the 90s on mRNA. It was easy to follow the literature because every year there was just one paper. But when I try to follow that what happens, many of these laboratories were not publishing more, not follow-up papers. For me, the most exciting was when it was shown that vasopressin mRNA could treat an animal, which was a Blatterboro rat, and which had some special disease, and injecting the RNA for 2 weeks, the animal was cured. So this gave me the idea that, you know, this mRNA can be used. By that time, I started in '89 doing some experiment, but this was very exciting for me. And you can see that there were publications on using mRNA for infectious disease against like influenza. And there were some papers where mRNA was used as a cancer vaccine. Only these scientists made follow-up and did a clinical trial, but most of the other groups stopped using. So what happened? What happened is the RNA had just problem. A very small amount of protein was produced, very short period of time, it degraded, and as we discovered also, it was immunogenic. So from the 90s, what happened is that continuous incremental improvement of the RNA, it purified a better cap, better process, and better formulation. That was happened. So everything was just incremental, and people usually they are not patient. And so, you know, many of the people left the field. So as I mentioned, I started in '89 working with RNA, and with my colleague, I started in cardiology, was interested in urokinase receptor. And what was interesting is for us, and why I am mentioning this example, because we generated an mRNA which coded for this urokinase receptor, and this protein, which is coded for this protein, the protein is highly modified, glycosylated, GPI linked, the carboxylator everywhere is modified. And what was interesting is we delivered the RNA inside the cell, and the cell knew what to do, how to decorate, and made it functional. So at that point, we thought that it could be useful for some kind of cell therapy. But Again, I have to leave this position. I have to say that I wouldn't be here if I wouldn't be terminated in my position several times. So now that I moved to neurosurgery, still at Penn, and I worked 17 years in this laboratory, and we tried to develop messenger RNA treating a patient who had a stroke. And interestingly, again, I work with a physician who visiting the patient, and I am in the laboratory, and we try to educate each other, find a common ground, and then develop something. So we did a lot of improvement here, but we were, again, was not aware of certain things which I learned when I met my fellow awardees, and Drew Weissman, and you will hear from him more. So that Drew was coming to University of Pennsylvania, and he wanted to develop a vaccine. I was not interested in vaccine, but I was interested to help and to work together. And Drew generated these dendritic, human dendritic cells. And when I made the RNA and he exposed to it, everything seems that the RNA is the perfect vaccine, as all of these markers were upregulated, and also found that inflammatory cytokines were increased. So it was not a good news for me. I was not very happy about inflammatory molecules. And we were thinking with Drew that what could be the reason, because the RNA, I assume that what I'm making is the same as inside the cell. So we thought that because this is immune cells, and maybe because it's coming the RNA from outside, it's recognizing as a danger. And to find out that whether if we isolate RNA out of our cells, whether it will be also immunogenic, similarly what I made in the tube. So for that, we did this experiment, and we isolated RNA from different compartments of the mammalian cells. Here is in purple what I synthesized in the tube, and you can see that what we measured on these dendritic cells is the inflammatory molecule called TNF-alpha. So I made the most inflammatory molecule, and what we isolated was not as inflammatory. Interestingly, the tRNA was not induce any TNF-alpha at all. Knowing that transfer RNA has a lot of nucleoside modification, this gave us the idea that maybe modification is needed to suppress immunogenicity of RNA. So we were thinking that, okay, what kind of modifications are there? And there were already a lot known, is present, more than 100 modifications in our RNA in the purine and pyrimidines. The only known modification, which enzyme, because we couldn't just order an enzyme to introduce and prove that really modification can suppress immunogenicity, the only modification was known 2'-methylation and pseudouridylation, and this was discovered by Tamás Kis, is a fellow Hungarian student from the same university, same dormitory. We lived and studied and together, and so I called up Tamás and asked help and told him that, can you send those things so that I can change and modify the RNA? And because I was nice to him, so he responded, but he said that he cannot send because really these things require special structural element, and I don't have in my coded RNA those. So then I call up János Ludwig and ask him that where I can buy nucleoside modified RNA, nucleoside modified triphosphate, because the other idea was that maybe we have to purchase those and then try to see that whether we can incorporate. So we purchased 10 of those, which is present in the human body, and we were testing out whether we can make RNA. And 5 of them, these incorporated to the RNA. So now that we had nucleoside modified RNA and ready to test that whether it is indeed is responsible for immunosuppression. What we found is that some of these were still immunogenic, some of these RNA, but others were not immunogenic, doesn't induce any TNF-alpha. When we made a closer look, we found that all of these actually modified uridine-containing So we concluded with Drew that if uridine is modified in the mRNA, then it is not immunogenic. Of course, we wanted to make whether vaccine or I want to make therapeutic protein encoding RNA, so that we needed that it will code for a protein. So the next step was to see whether it can be translatable. And well, 2-thiouridine modified was not translatable at all, but when we use the pseudouridine containing RNA, we get 10 times more protein on it. So it was now is a very good news that we get non-immunogenic RNA, which can be translated. I have to say that we describe also that TOL7 and TOL8 was actually activated uridine-containing RNA, and later, 10 years later, it was demonstrated crystal structures that actually the uridine, the nucleoside itself, can help to dimerize toll-like receptor 8 and activate it. And also when we did with experiments, we didn't know that our mRNA has pseudouridine, but turned out that again, 2014, several papers published that our RNA also has pseudouridine in it and incorporated by different mechanism. What we have done, we did after all of our discovery, what usually in the United States you do, you file a patent, then send in the paper, and then establish a company. We established RNARX, and we submitted also a small business grant, and we proposed that we will deliver to animals erythropoietin-coding RNA, and we will demonstrate is that it can increase the hematocrit of those animals. And that was a proposal for treating anemia with mRNA. So we generated mRNA, as coding here in the red, shown pseudouridine-containing RNA, and uridine-containing RNA, and we also injected just erythropoietin to the animals. And this protein half-life is very short, so it quickly disappeared. But what you can see here that pseudouridine-containing RNA, when we injected and translated to erythropoietin, we could measure the protein for 4 days. So it was extended translation of this molecule, which otherwise has 2 hours half-life. And it was physiologically relevant, and it was biologically active because it increased the hematocrit. And when weekly we injected, then we could maintain that high level of hematocrit in the animals. And it was important to demonstrate that pseudouridine-containing RNA even in the mice was not immunogenic, did not induce interferon. So this was the point, again, that I had to change, was forced to change my position, and then ended up in Germany at the BioNTech. At that point, we were not in this fancy building, we were on the campus, and BioNTech had no website. I just accidentally learned existence of it, but I went there and joined forces with Ugur Sahin and his wife, Özlem Türeci. And first we wrote a big paper about what you should know about mRNA, and we further improved the mRNA. You can improve the RNA by codon optimization, purification, nucleoside modification, many things. So as a result of all of our work we did with Drew, we did here in BioNTech, instead of this small amount of protein, short period of time, now we could make a lot of protein for extended time. So the mRNA was ready for prime time and test out. And my first project at BioNTech, we did messenger RNA. This contained 1-methylpseudouridine and coded for bispecific antibodies, recognizing one part is the immune cells, the other part is the tumor cells, and bring close proximity. We could demonstrate that such bispecific encoding mRNA injected into the mice can eliminate already established large tumors. These studies is presently in human trial. The other project we did when we injected mRNA to the mice, and this was again a cell surface closed tumor, so that's like this is also in human study for head and neck cancer and melanoma, and these tumor were injected with mRNA coding for these cytokines, and these made the cold tumor hot. Immune cells rushed there, and then when it circulated, they could eliminate the remotely located tumors, so that when it was injected these mRNA coding these cytokines, the lung had from the lung eliminated these tumors. So while we were working on this project in BioNTech in Germany, my colleague Norbert Pardy in Drew Weissman's lab generated vaccines using nucleoside-modified RNA. This was 2017, and that time the Zika virus was a major problem, and what Norbert was doing, generating mRNA coding for this envelope encoding protein of the Zika virus, and this was 1-methylpseudouridine, the nucleoside-modified RNA. The formulation was by Acuitas, the same as was for the BioNTech Pfizer vaccine, and of course, we know that, and you will hear from my fellow awardee, Peter Kullis, about importance of the lipid nanoparticle, which contains different elements and were important adjuvant activity. And what happened is when these were injected to the monkeys, very small amount could protect the monkeys from Zika viral infection when it was challenged. So even 50 microgram it was very revolutionary, because I have to say that prior to that, all of these DNA vaccines which were used unproportionally had to give higher dose to when the animal size was large. And so in human case, it was already milligrams had to be injected, but not in the case of mRNA. So this paper actually was selected for BIAL, the most important paper in the last 10 years. And so this was served the basis for developing the vaccine. And you are already quite familiar how we, 2020 January, we learned the sequence information. And by the end of that year, we also get the vaccine. So if all of these things would happen even just 20 years before, information would not have been enough to receive, because 20 years ago, you just couldn't order a DNA synthesis, so the genes couldn't be ordered. But here you could order, and of course, with the help of input from government and others, it accelerated and helped to run the clinical trials. And that, of course, many people thought at that point that this was the first time nucleoside-modified mRNA was injected in human. But you might not be aware of that, that 2018 messenger RNA, nucleoside-modified RNA, was already used in a human trial for heart failure, patients who are undergoing bypass surgery. The RNA in 30 different point was injected to the human heart, and that RNA coded for VEGF-A, which is a protein that generates new blood vessels and then increase the blood flow in a part of the heart. And this was already in phase 2 trial and was very successfully completed, and the patient who received the mRNA felt much better. And other trial was, which was again using nucleoside-modified mRNA by Intellia. This was a Cas9 mRNA, and this was treated for patients who get amyloidosis, the disease. Here, the mRNA coded for Cas9 protein, as well as the guide RNA was delivered in the patient infused, and this toxic protein made in their liver cells. And so what happened is, with this technology, the genes where the toxic protein was produced was interrupted. And it was only one injection was sufficient that a patient, even 1.5 years later, were not producing any toxic protein. So this was very successful and ongoing. And right now, I don't talk about the vaccines, but for vaccine messenger RNA, nucleoside modified RNA is used for now that not just for COVID-19, that's approved, but there are CMV, RSV, other viruses, HIV, and influenza, and so on. But I was always interested in the protein replacement therapy when the mRNA code for therapeutic protein. And those mostly, of course, secreted products were tested at the beginning, and this were the VEGF-A. Monoclonal antibody, BioNTech has one, not just a bispecific antibody, but one mRNA recognizing cancer-specific proteins, and cytokines, which was also injected in the, whether in the tumor or otherwise. And a lot of preclinical, I don't even have time updating all of these because so many trials, so many new companies are formed and proceeding with that. And I am very grateful for my colleagues who I work with at BioNTech, mostly because prior to that, I never get an R01, so I never cannot say thanks to the graduate student and the postdocs because I didn't have one. And so most of this experiment I did with my own hands. And I am also very grateful to my family. My daughter is a 2-time Olympic champion rower. So I am telling to the girls here that, you know, you don't have to choose whether you have a career or a family. You know, here I am, an example. And thank you very much for your attention.
Fantastic. Thank you so much, Dr. Carrico. Phenomenal talk. So good morning, everyone. I am Professor Leah Cowan, Vice President, Research and Innovation and Strategic Initiatives at the University of Toronto, and I'm thrilled to join you as the chair for the rest of session 1 today. Our first early career investigator speaker is Professor Anna Blakeney, who is Assistant Professor in the School of Biomedical Engineering at the Michael Smith Laboratories of the University of British Columbia. Professor Blakeney was selected by Drs. Peter Cullis, Catalin Carrico, Drew Weissman for her next generation RNA vaccines and therapeutics research. And I am absolutely delighted to turn it over to Professor Blakeney. Over to you.
Excellent. Well, it is truly my honor to be here today. So thanks to Gairdner and Kati and Drew and Peter for selecting me. It's really a really exciting opportunity for me. So today I'm going to tell you a little bit about my work at University of British Columbia. First, I'd like to acknowledge that I live and work on this beautiful campus in Vancouver in BC, which is the traditional, ancestral, and unceded territory of the Musqueam people. And obviously, it's been a really exciting time to work in the field of RNA, as Kati so beautifully set me up for this presentation. And so I like to say the plane is off the ground. You know, a few years ago when I would get talks, I would have to tell everybody what RNA vaccines were and try to convince them that they're working. But I think obviously we've seen it's come a long way in just the past few years. And this is really due to the efforts of Drew and Kati and Peter. They've really set the stage for the work that I and many others are doing now in the field of RNA vaccines and therapies. So where do we go from here? So my lab at UBC really focuses on a few different aspects of this RNA research. So we work on RNA molecular engineering. So how do we design better RNA that results in higher amounts of protein expression? Of course, the biomaterials design. So how do we get these large negatively charged molecules into cells more efficiently and into the cells and tissues that we want the therapeutic to be in, as well as thinking about better vaccines and other applications for this technology. So other vaccines such as bacterial antigens, gaining an improved understanding of the immune response, as well as expressing monoclonal antibodies, which could be used for treating cancer or preventing disease. So we've all are very well aware now that the safety and the efficacy of the mRNA vaccines have been very well proven against COVID-19. So this is some of the data from the Pfizer and BioNTech trial, which really, I think, blew everybody out of the water as far as how effective it was. I'm not sure anybody was quite expecting it to be as high as effective as 95% protection with just the 2-dose regimen. And we've really seen that, compared to the placebo, we have a really high efficacy rate, which is super encouraging for those in the field, as obviously before COVID-19, nobody had ever done a phase 3 trial for RNA before. In addition, the safety over a median of 2 months from this paper that was published initially was, similar to that of other viral vaccines, and so it got this glowing report, and obviously now billions of people have taken these vaccines. So where do we go from here? So one of the priority areas in the field is really thinking about ways to reduce the dose of RNA. So this is actually some data from one of Moderna's clinical trial where they looked at using 3 different doses of RNA, so 25, 100, and 250 micrograms. And there's a lot of data here, but you can see that they've categorized the side effects as mild, moderate, and severe. And what's very visually apparent from this data is that as you increase the dose of RNA, you're also increasing the frequency and severity of the side effects. So we now understand that the dose of RNA is directly correlated to the frequency and severity of these side effects, and this means that minimizing the dose of RNA is really required for use of mRNA vaccines in non-pandemic contexts, as well as for other applications such as therapeutics. So one of the ways we're able to do this is with what my lab works on, which is called self-amplifying RNA. So at the very top, we have the structure of a conventional mRNA. As Kathy said, we have the 5' cap and a 3' poly A tail, as well as an untranslated region at the 5' and 3' ends. For self-amplifying RNA, many of the structures are very similar. We have a 5' cap and a 3' poly A tail, as well as a 5' and 3' UTR. But we also have these 4 non-structural proteins, which are typically derived from an alpha virus and make up a replicase. So what this means for the self-amplifying RNA is that it's actually able to make copies of itself once it gets into a cell. So on the right, we have a typical mRNA. Once it enters a cell, it engages with the ribosome, and you get translation of your antigen. Self-amplifying RNA starts out very similarly. Once a copy gets into a cell, it engages with the ribosome. You do get some translation of your antigen from that initial engagement, but it also translates its replicase. So the replicase goes back to that original strand and makes many exact copies of it. For the sake of this very simplified schematic, I just have 3, but it actually makes thousands of copies in the cell. And so this results in a much higher level of protein expression from the same initial dose of RNA. So what this means is that typically we're able to use about 100 times less saRNA compared to traditional messenger RNA. So one of the things we really think about is how does the innate immune system sense these RNA and the delivery vehicles, and how can we really engineer ways around this? So this is from a review that Norbert Party wrote in 2018, and I always think back to this for a number of reasons. One is, we know all these ways that RNA is sensed in the cell, right? So endosomal sensing with TLR3, 7, and 8, and cytosolic sensing with some common ones like MD8-5 and RIG-I. But on the right, we have this idea of carrier sensing, and there's just a big question mark. So we have a pretty good idea of how the RNA is sensed, but we're still learning a lot about how these delivery vehicles are sensed by cells. And this is a lot of the work that Kati and Drew have done, is thinking about ways that we can navigate around this type 1 interferon sensing, which is kind of a double-edged sword for RNA. So it leads to a good vaccine immune response, but it can also completely shut down the protein translation within the cell. So one of the things we have done, and I'll just tell a very short story about our research today, is think about incorporating these virally derived interferon inhibiting proteins directly into our RNA. So at the top is our wild type replicon or saRNA, and at the bottom we screened through a number of interferon inhibiting proteins or IIPs. So many viruses have naturally evolved to have these two because they have the same issue introducing foreign RNA. So one of the things we did was to screen through this, and it's encoded directly in the same RNA, so we guarantee that this protein is always being expressed in the cell where we have the RNA. So one of the things that we saw from this is we were actually able to screen this in human skin explants. And as anybody who's worked with RNA has probably observed before, is that as you increase the dose, it doesn't have a linear dose response. It actually has a parabolic dose response where once you get to a point where further increasing the dose does not increase the protein expression that you see. So in this case, we were looking at kind of our two lead candidates. So our EGFP is just our wild type, and then PIV5 and MERS-CoV2 or F4A. And in order to look at the protein expression, we looked at the percentage of GFP positive cells at 3 different doses of the RNA. So 0.2, 2, and 20 micrograms. And you can see all the groups are about similar for the 0.2 and 2 microgram dose, and there's a slight increase in the number of GFP positive cells. But as we go up to the 20 microgram dose, we see the wild type drop off, and we actually have even fewer GFP positive cells than the 0.2 microgram dose. Whereas for our siRNA that has the interferon inhibiting proteins incorporated into it, we actually see a higher number of GFP positive cells, telling us that by inhibiting this interferon pathway, we're actually getting good protein expression with increasing the dose. So we use this also in a vaccine model to see, we're seeing more protein expression, but does this also increase the vaccine immune response that we're seeing? So we did this in this case with a rabies vaccine. So on the left, we looked at the antibody titers, the IgG, as determined by ELISA. And on the right, we looked at the neutralization. And in this case, we were using just one of the IIPs, which is called ORF4A. And the rabbits got 2 injections in this case at 0 and 4 weeks. And we looked at the antibodies at 4 and 6 weeks after the injections. And you can see for the wild type group that we get a good antibody response, about 10 to the 4th nanograms per mil. But with our IIP, we get almost an order of magnitude higher antibody response, indicating that not only is this yield increased protein expression, but also a better vaccine immune response. So one of the things that I'm really passionate about as well is science communication, so I just wanted to touch on this a little bit. But in January 2020, there were still very few scientists that knew about the RNA vaccines, and now billions of people have gotten them. And I think many of us can attest to that there's been quite a PR problem for the RNA vaccines over the pandemic. And so this is one of the things that I'm trying to change. So I started a TikTok channel that's dedicated to educating people about vaccines. And I think something that I've noticed is that there's just a significant gap between the research that happens and what the public knows about. And so I think as scientists, we can all do a better job of communicating exactly what we're doing so that people aren't surprised by these technologies when they come out. So one of the things I've done is Katty, Drew, and Peter have all been, I'm not sure if it's honorees or victims of the TikTok channel, but I've tried to highlight all the work that they've done just because I think people really appreciate scientists, but they also have no idea who they are. So part of my mission is to make more scientists household names. And you can see from the comments on the right, this was from one of the videos with Peter, that people are so appreciative. They say, like, so grateful to you people, and I would love to get it. I just got my 2nd dose an hour ago. And so there's really been kind of an amazing recognition, which has been really cool to see. So with that, I just wanted to conclude that we've seen these mRNA vaccines have completely revolutionized the way that we're able to respond to pandemics, in part from contributions from Katty, Drew, and Peter. I think for the next generation RNA, thinking about modulating this innate sensing in the immune response is imperative to improving these, both on the vaccine side and using these for therapeutics as well. And for the science communication side, I think it's important to keep in mind that public engagement really helps to close the gap between the research and what the public is aware of, which helps when we try to implement these technologies. So with that, I'd like to thank everybody in my lab and collaborators, as well as Gairdner, for having me here. It's really my pleasure, as well as our funding sources for the lab. And with that, I will take any questions.
Thank you so much, Professor Blakeney. That was fantastic. So it is my pleasure now to introduce Dr. Drew Weissman. Dr. Weissman is one of the recipients of this year's Canada Gairdner International Award for his pioneering work with Dr. Carrico in developing nucleoside-modified mRNA therapeutics, of course, the foundational technology for the highly effective COVID-19 mRNA vaccines that we've been hearing about. He is the Roberts Family Professor in Vaccine Research, Director of the Penn Institute for RNA Innovation, and Director of Vaccine Research, Infectious Diseases Division at the University of Pennsylvania, Perelman School of Medicine. So I am delighted to turn it over to Dr. Drew Weissman. Thank you.
Thank you very much. So I'm highly honored to receive this award. We've been treated great in Canada, visiting all ends of it, from the sounds of it, of all the other awardees. What I wanted to do in today's talk is to give you an idea of the potential breadth of RNA technologies in the future. Hopefully everybody in this room has received a vaccine in their arm, but there's huge potential for what RNA can do in the future. Let me just start by telling you the difference between RNA vaccines and all the other vaccines made. Making an RNA vaccine is very simple. You design the sequence, Katie described this. You in vitro transcribe it, that's a 2-hour reaction. You purify away the contaminants. You mix them with lipids that forms nanoparticles. You get rid of the ethanol and salts, and you put it in a vial, and you've made a vaccine. If you want to change the vaccine, you change the sequence design, everything else is identical. For all other vaccines, when you change the vaccine, when you make a new vaccine, you start over. You figure out how to produce the protein, how to purify it. Everything is different. Everything is a huge amount of work. So when we started looking at vaccines, we looked at influenza. This is just a review of the current literature when we were first investigating influenza vaccines. The first two are similar to the human vaccines, the live virus we give to kids, the inactivated virus that we give to adults. And what's in parentheses are the viral titers you see in mice after 2 immunizations. It's called an HAI titer. It's a pseudo-neutralization titer. But you can see with all of these, titers in the 1 to 100 range are typical. 1 to 40 is protective, so this is considered a good titer. When we looked at HA encoding mRNA, and we're now comparing it to a live virus or inactivated virus, the titer from mRNA was 1 to 2,500. That's 50 times better than an activated virus, 5 times better than live virus. This excited a lot of biotech and pharmaceutical companies, because typically in vaccine research, if you can double a titer with a new vaccine, people are excited. But as an immunologist, we wanted to understand why this vaccine was working so well. So this is a cartoon of how a B-cell response develops. Follicular B cells interact with particles, viruses, antigens, adjuvants. They get help from CD4 positive T cells and form germinal center B cells. The germinal center is the site where the B cells rapidly proliferate, affinity mature, and form plasma cells and memory cells. So we measured all of these populations. We're measuring the number of antigen-specific B cells of each type. We have a fluorescinated HA in the flow cocktail, and then other antibodies that identify each of these 3 populations. What you can see is that in all of them, we have about a 50-fold increase in the number of antigen-specific B cells. That accounts for the increase in antibody titer. We left some mice for over a year and went back and we measured the long-lived plasma cells in their bone marrow. Again, we saw huge numbers of these cells still present after a single immunization. But that didn't explain the mechanism, and we wanted to understand why we were getting these huge antigen-specific B-cell responses. It came down to what are known as T follicular helper cells. These are a form of CD4 helpers that form germinal centers, and they supply the help to the germinal center B cells in order for them to affinity mature, proliferate, and make memory and plasma cells. We used a macaque model system, and in these macaques, we gave them either an mRNA that encoded the HIV envelope, or we gave them that same envelope protein with a double-stranded RNA adjuvant. We chose that because that was one of the best described adjuvants for inducing TFH cells. But as you can see, there's no comparison to the effect of the mRNA LNPs and the double-stranded RNA adjuvant. Huge numbers of TFHs were induced by the mRNA LNPs. We did a calculation. Typically, for most vaccine adjuvants, double-stranded RNA, alum, MF59, 5 to 10% of the CD4 helpers are TFH phenotype. For mRNA LNPs, it's over 50%. So this is an adjuvant that selectively induces TFH cells that drives B cell responses. That's what accounts for the huge antibody responses. We wanted to test these in an animal system. So in influenza, every year you have to make a new vaccine because the head of the virus, the HA, mutates and prior vaccines don't work. We challenged the animals with a different subtype of HA. This is from an H5 avian virus that occasionally causes pandemics. We saw complete protection with an H1 vaccine against an H5 virus. This is now going forward into humans as a universal influenza vaccine. I want to switch topics and now talk about the future of RNA therapeutics. If you've worked in this field, you know that the critical difficulty is targeting, is getting the nucleic acid or the virus to the cells of interest. With LNPs, they go to the liver. They bind ApoE and they target hepatocytes. They also go to dendritic cells, which makes them a good vaccine. We wanted to know, could we target LNPs to other cell types? We concentrated on endothelial cells, T cells, and bone marrow stem cells. The platform that we used was to simply change the surface of the lipid nanoparticle. We attached antibodies, pieces of antibodies, receptor ligands, other proteins to the surface of the lipid nanoparticle using different types of conjugation chemistry. When we did that, we saw that we didn't change the morphology of the LNPs. They got a little bit bigger, which is the addition of an antibody coating, but they retained their surface charge. And most importantly, they retained function. So you can see in C, these are UVECs, primary human vascular endothelial cells. In the absence of targeting, you get no delivery of LNPs or mRNA. With targeting, we're using a PCAM antibody to target, and PCAM is expressed by UVECs. We see high levels of uptake and translation of the mRNA. So we could selectively target human endothelial cells in culture by targeting them with a PCAM antibody. We injected these LNPs into mice, and what you can see are really two things. The first is that when we added an antibody to the surface of the LNP, we reduced liver uptake. We showed that we're actually blocking ApoE binding. Most importantly, we're now seeing lung uptake, and about 50% of the delivered LNPs are going to the lungs. Lungs have the highest level of PCAM on endothelial cells. We wanted to see what else could we use this for. So in the cerebrovascular elements of science, strokes are a common occurrence. Typically, when a patient has a stroke and you make a CAT scan 24 hours later, you see a small area of dead brain that's fed by that middle cerebral artery that had the stroke. But that's not the main problem of a stroke. What happens in the following days is the stroke extends and gets much larger. And that's because that initial stroke leads to edema, inflammation, and extension of the area of brain death. So we developed a system where we could inject TNF-alpha, a pro-inflammatory cytokine, into one hemisphere of a mouse brain. And then we treated those TNF-inflamed brains with a nanoparticle that targeted the brain, targeted the blood-brain barrier using VCAM targeting. We delivered an mRNA that encoded thrombomodulin, which is an anti-clotting, anti-inflammatory protein. And what you can see on the left is that when we treated animals with a VCAM-targeted LNP with thrombomodulin, we got rid of all of the vascular leakage. So that in theory, would get rid of that extension of the infarct to greater portions of the brain as a potential therapeutic. My lab has had an interest in HIV for a long time. So we wanted to see, could we target T cells? We did the same thing. We added antibodies to the surface, and we could successfully target primary CD4 positive T cells. But most importantly, if you've ever worked with T cells, they don't have endocytic activity. That means they can't endocytose free particles. You can't deliver nucleic acids with lipids, polycations, or other particle delivery systems. The only way you can do it was electroporation. Our hypothesis was, if we had an antibody that bound an antigen on T cells that was endocytosed after binding, that would bring the lipid nanoparticle into the T cell. And that's what we're seeing here. Purified human T cells targeted with ibalizumab, which is a clinically used anti-CD4 antibody, gives high levels of luciferase activity. The T cells are taking up the LNPs, the RNA is released, and it's translated. When we inject these into animals, the spleen lights up. But most importantly, we re-imaged those animals after we took out all of those organs. And what we're seeing now are lymph nodes. These are para-aortic and inguinal lymph nodes are lighting up. So the LNPs injected intravenously are getting out of the circulation, into the tissues, into the lymphatics, to the draining lymph nodes, and they still have the capacity to bind T cells, deliver the mRNA, which is translated and shows up. We switched to a model system that has a LOXP reporter gene in it, in this case, a green fluorescent protein. The mRNA that we delivered with the LNPs encoded Cre recombinase. So the Cre recombinase removes the stop codon and makes targeted cells green. We injected the targeted Cre recombinase LNPs into an animal, and without targeting, you get very little green protein. With CD4 targeting, you get high levels of green protein in lymph nodes and the spleen. But what's important to notice here is that on the y-axis, that's an activation marker. So we're seeing equal amounts of delivery of LNPs to resting T cells as we see with activated T cells. That's important because in HIV, the resting CD4 positive T cells are the site of HIV latency, and they have to be targeted in an animal or in a human. We also wondered, could we deliver CAR T cells? So this is what a CAR T cell is. In CD8 positive T cells recognize antigen plus MHC. And when they recognize that peptide MHC, they kill those antigens on a tumor cell or a virally infected cell. An alternative was developed known as a chimeric antigen receptor. And what happens there is that the CARs recognize a specific antigen. And the first that were developed were CD19. CD19 is expressed by leukemias and lymphomas. And CARs that recognize CD19 delete the tumors. They have clinical efficacy in the 60, 70, 80% range. That's a huge advance in cancer chemotherapy using the delivery of CAR T cell. The problem is, this is what it takes to make a CAR T cell. You have to take cells out of a patient, you infect them with a lentivirus, you expand them for a couple weeks, and then you give cells back to the patient. It works great, but it costs $500,000 per dose, which is problematic. What we proposed is whether we could make CAR T cells in vivo. So our idea was that if we could target T cells, and in this case, we used a CD5 targeting molecule that targets both CD4s and CD8s, and encode the chimeric antigen receptor in the mRNA. The T cell would take up the LNPs, the RNA would make CARs on the surface, and we chose a CAR that targeted fibroblasts. We tested this in vitro. 80% of the T cells made the CAR. In vivo, 25% of the T cells express CARs on their surface. And we used a model of fibrosis. So if you make a mouse hypertensive, they develop cardiac fibrosis. Most cardiac diseases are mediated by the fibrosis that forms after the disease insult. This is an echocardiography, a way of taking pictures of the function of the heart. And we can see on the right, we're measuring diastolic and systolic volumes. When the heart becomes fibrosed, it can't function, and the volumes get bigger. In the bottom right is the ejection fraction. This is the amount of blood that the heart pumps out with each beat. When the heart becomes fibrotic, the amount of blood that's released is reduced, and the ejection fraction is reduced. So gray is a normal animal, yellow is a fibrotic animal, red is those animals treated with one treatment of CD5 targeted LNPs that make FAP CARs. And we can see we return their cardiac function to normal. When we stain the hearts of these animals, the normal saline treated on the right, the middle are fibrotic animals. You see high levels of fibrosis. The left is one treatment with FAP CAR LNPs targeted to T cells, and we return the fibrosis to nearly normal. So with an off-the-shelf mRNA LNP, we could cure cardiac fibrosis in this animal model. Another thing we were interested was targeting stem cells. So bone marrow stem cells are the source of all of our red cells, white cells, granulocytes that allow our immune systems and our respiratory systems to function. Sickle cell anemia is a disease that has 300,000 people born with it every year. They're mainly in Africa, but you see them across the world. The problem is you can't do bone marrow biopsies and viral infections on 300,000 people a year in regions like Africa and India and the Middle East. It costs about $1 million per treatment, and the facilities to do these things aren't available. So our idea was that if we could find antigens that selectively targeted bone marrow stem cells, we could deliver LNPs and target those. And that's what we did here. We saw that with a single treatment, 85 to 95% of bone marrow stem cells and progenitor cells were targeted by the LNPs, and we could mediate gene recombination in these cells. Most importantly, what you need to do is called a secondary transplant. So you treat mice once with the LNPs, you wait 6 months and take their bone marrow and put them into irradiated mice that no longer have bone marrow stem cells. And we found that our targeted LNPs from the previous mice, 100% of the red cells, white cells, and granulocytes were gene recombined. So that means that with a single injection, and in this case it's 0.1 micrograms of mRNA LNPs, could gene edit 100% of the repopulating bone marrow stem cells. So now we can go to Africa, India, the Middle East, the United States, Europe, anywhere there are people with sickle cell anemia. We can give them a single injection of mRNA LNPs that target the bone marrow and that can correct the sickle cell gene defect and cure their disease. So instead of $1 million per treatment, now we're looking at, I don't know, a couple $100, maybe $1,000 per treatment. And we can easily cure sickle cell anemia with a single injection intravenously of RNA LNPs by targeting them to this bone marrow stem cells and delivering an enzyme through RNA that fixes the gene defects. You can do a lot of other things. You can deliver toxic genes to stem cells. So now when we do bone marrow transplants, you have to irradiate people or give them high doses of chemotherapy to kill the bone marrow stem cells. The problem is it also kills the gastrointestinal cells and many other necessary cells. That has toxicities and mortality up to about 5%. Now we can deliver an mRNA LNP. We can selectively kill the bone marrow stem cells and easily do transplants. We can also deliver therapeutic proteins that stimulate B cells, granulocytes, platelets, many other cell types that can be easily delivered to the bone marrow. So our conclusion are that mRNA LNP therapeutics has the potential to do many different things. We have vaccines for many different diseases in clinical trials. We're developing ways of doing gene therapy in bone marrow stem cells. We can target heart, we can target brain, we can target lungs. That allows us to deliver mRNA LNP therapeutics to many different organs and cell types. And we see this as the future of mRNA LNP therapeutics. I need to thank all of the people in my lab and all of the labs that we collaborate to do these. That includes Katie, who together we developed nucleoside-modified mRNA that led to the vaccines for COVID currently in use. It included Acuitas Therapeutics, which is a company that Peter Cullis started and who we've worked with for 10 years or so to develop LNPs as an mRNA delivery platform, and my other collaborators for all of these studies. Thank you.
Thank you, Dr. Weissman. Just fantastic work. So our next early career investigator lecture is from Professor Bowen Lee, an assistant professor in the Department of Pharmaceutical Sciences and the Institute for Biomedical Engineering at the University of Toronto. Dr. Peter Cullis, Catalin Carrico, and Drew Weissman selected Prof. Lee for his work on lipid nanoparticle technology. Delighted to hand it over to you, Professor Lee.
Good morning, everyone. So thank you for having me here today. First of all, I want to thank my heroes, Dr. Peter Cullis, Dr. Carrico, and Dr. Weissman for selecting me for the ECR award this year. So my first slide, I want to recap the contribution of this year's Gairdner Award Laureates. So Dr. Carrico and Dr. Weissman basically teach people how to safely place the chemically modified mRNA in human body for therapeutic purposes without causing any trouble, like inducing any inflammatory responses. Meanwhile, Dr. Peter Kless spent almost half a century to teach people how to deliver this kind of nucleic acids. The ionizable lipids, ionizable lipid nanoparticle enable us to safely use this amazing chemically modified mRNA. So without their contribution, we won't be able to save the COVID-19 mRNA vaccines, which have saved millions of people's lives. So the new technology of mRNA vaccine actually, revolutionized a whole field of vaccines. It makes the development process of vaccine much easier than before. During the pandemic, the scientists were able to easily sequence the spike protein on the coronavirus, make the mRNA using templates from the spike protein, and load them into the ionizable lipid nanoparticles developed by Dr. Peter Kless. And then once this mRNA vaccine or mRNA loading nanoparticle got injected into the human body, they can in situ express spike protein in human cells, and then which can induce protective immune responses. So previously, before the mRNA vaccine technology, it usually takes 5 or 10 years for human to develop one vaccine. But the mRNA vaccine was developed within 12 months, so which actually significantly improve, the protection of human health. Now, when we have this amazing or magic molecules, how can we deliver them? Or we call it the hopeful message. How can we deliver this hopeful message? The mRNA molecule itself is very big compared with a lot of small molecule drugs. It's about 1,000 kilodalton. Meanwhile, they are extremely unstable and they are very vulnerable to nuclears in human body. And more importantly, they only work in the cytoplasm, different from many small molecule drug or antibody drug. So that's why we need the lipid nanoparticles. The classical compositions of lipid nanoparticle composed of the 4 ingredients, among which the ionizable lipids plays the most important role that can stabilize the mRNA, meanwhile allow the mRNA to escape from endosome after being taken up by the cells. So this scheme basically shows how does the lipid nanoparticles work. Once the nanoparticle is taken up by the cells, the ionizable lipid will play a magic that can break the endosome membrane, allow the mRNA get released into the cytosol, which will later on bind with the ribosome and lead to the protein expression, which can later on be presented by MHC2 or MHC1 to induce corresponding immune responses. So these lipid nanoparticles play a super important role in the development of mRNA vaccine. So that's why the title of the breaking news of chemical engineering news say that without these lipid shells, there will be no mRNA vaccines for COVID-19. So this picture basically shows what is the landscape of all of the RNA LNP drugs that have already been approved for clinical use. So the first one is the first siRNA drug, which is another type of RNA medicine that was approved by the US FDA in 2018. The other two, you must be familiar with the other two. One is Moderna's mRNA vaccine. The other is the mRNA vaccine from Pfizer and BioNTech. The reason why I want to put them all of the formulation together here is I want to emphasize, as you can see that except for the ionizable lipids, all the other ingredients in these formulations are almost the same. So that means that actually the ionizable lipids is the core of the LNP technology that differentiates different companies. So my lab basically focuses on streamlining the development of next generation lipid nanoparticles using high throughput technologies. Because the one-size-fits-all approach actually doesn't work for RNA delivery. In order to maximize the mRNA potential for more disease in other organs, not only for muscle, not only for brain, not only for bone marrow, we really need to identify or expedite our process to identify new nanoparticles to satisfy the delivery tasks. So basically, our group has established a multi-component reaction that can make the process of synthesizing ionizable lipids much easier than before. So we can use this one-port multi-component reaction to prepare thousands of ionizable lipids within 1 day. After synthesizing this combinatorial libraries of ionizable lipids, we will formulate them with the other ingredients, lipids ingredients into nanoparticles. and then perform a high-throughput screening on this lipid nanoparticles to identify the one that can satisfy our needs. So here, I just want to show one small cases, like how can we leverage our platform to identify new nanoparticles to find new application for mRNA therapy. So inhalation of mRNA for treating lung diseases has been receiving more and more attention. So if we can help people to inhale these mRNA therapies, we can treat a lot of lung-related disease, such as cystic fibrosis, like AAT disease, lung cancer, or COPD, or even COVID-19. However, the delivery to the lung has been more challenging than the muscle due to the existing of mucus and other biological barriers. So for example, one company called Bio, which has been focusing on lung delivery of mRNA for many years, they failed that clinical trial for cystic fibrosis treatments using mRNA. So that's why this company sponsored our work when I was a postdoc at MIT. And we use our high-throughput platform to identify new lipids for them, specifically for the lung delivery of mRNA. So as I say, that we are highly interested in the multi-component reaction. Again, here, we classified the structure of lipids into 3 parts: head group, linker, and tail. And then based on this classification, we designed the structure for each component and simply mixed them together. So we were able to synthesize 720 lipids in this, for this batch. And then, luckily, after we performed the high-throughput screening, we identified one lipid. It's called RCB4-8 that is super potent in transfecting the lung cells. As you can see that when we compare this new lipid with MC3, which was another lipid approved by the FDA for RNA delivery, the RCB4-8 new lipid is almost more than 100-fold better than this benchmark lipid. And furthermore, we test whether we can use these lipid nanoparticles to deliver mRNA for gene editing in the lung. In this experiment, we used the AI14 mice. This genetically modified mice has a very unique gene sequence. So it contains a TD tomato signal in its sequence. However, we can't see any fluorescence from TD tomato in the normal cells due to the presence of a stop sign in its gene. So once the mRNA, the Cray recombinase mRNA is successfully delivered to a specific cell, this Cray recombinase will be able to remove the LOXP flanked outside of the stop sign and enable the TD tomato fluorescence to be seen on the specific cell. So in this way, we can easily tell which type of cell got transfected and how many percentage of the cells can be transfected in the airway. And we saw that we were able to very effectively transfect the lung epithelial cells. And we are extremely interested in lung epithelial cells because lung epithelial cells are actually the disease location for a lot of lung disease, particularly for cystic fibrosis. We're pretty excited to say that we can not only transfect it or edit the CD80 cells, but also club cells, which are kind of stem cells. That means that by transfecting or gene editing on the club cells, which are stem cells, we can provide a more like a long-term effect because after the CD80 cells died, the club cells can still differentiate into the CD80 cells. So finally, I want to thank Dr. Carrico, Dr. Weissman, and Dr. Peter Claes again for selecting me as the ECR awardee this year. And I also want to thank U of T and all the funding agencies that support our work. I also want to thank my colleagues and my collaborators at U of T and MIT and University of Massachusetts and Northwestern University. And particularly, I want to thank my lovely lab members. Okay, thank you so much.
Fantastic. Thank you, Professor Lee. So next, we have up Dr. Peter Cullis, who is one of the recipients of this year's Canada Gairdner International Award for his pioneering work on developing lipid nanoparticle drug delivery systems used in the COVID mRNA vaccines, the foundational technologies for the highly effective COVID-19 vaccines. Dr. Cullis is professor in the Department of Biochemistry and Molecular Biology at the University of British Columbia, and I am delighted to turn it over to Dr. Cullis.
Okay, I'm going to give a bit of a retrospective. You'll notice my voice is not that great, but I have been tested for COVID-19 and I'm negative. So, you know, that was a great relief, by the way. Coming all this way and not being allowed to speak would have been a problem. Okay, what I want to do today is really indicate to you that where you might see science as a linear pathway. It's not. We go through various iterations, and then things happen that are, you can only call it serendipity or good luck, and so I'm going to emphasize some of those aspects here. I'm also going to, emphasize the length of time sometimes it takes. This is literally 50 years of lipids for me. So, okay, I have a number of conflicts of interest that are indicated there. So I'll go back to the beginning. This was a long time ago, 1972. I got a PhD in physics using magnetic resonance. It seemed to me that the most interesting problems were well outside the field of physics. So I got, simply because I was an experimentalist, and if I did experiments and I showed something was different from established theory, it was very likely my experiment was wrong. So I got interested in the life sciences and was awarded by some miracle, actually, that I was awarded an MRC at that time, a postdoc fellowship to go to biochemistry department at Oxford. I knew absolutely nothing about biochemistry. I didn't know what DNA was, didn't know what a protein was, etc. So when I got to Oxford, I helped build an NMR machine. The lab there, they just moved from another lab. They had a big magnet, but they didn't have the electronics to go with the magnet for the NMR machine. So actually my physics background came in pretty handy, as I had to build this thing. So me and a graduate student put it together. And so it was, then they say, okay, well, what was I gonna do with this, apparatus? So what we decided to do was to use the NMR machine to study lipids in biological membranes. And this is something that once I started on, I've never stopped. So here you're seeing a picture of a biological membrane on the left-hand side. It's completely dependent on the lipid bilayer. I'm being exaggerating, of course, but very often we get, we take the view that the proteins, oh, thank you, I'm sure I'm going to need that at some point, that proteins do everything. They do all the work in biology, but lipid bilayers are pretty darned important. So all biological membranes, of course, have a lipid bilayer. And the membrane lipids are really quite amazing, and they have amazing properties of self-assembly. It's due to their amphipathic, as it's termed, structure, a polar head group and the hydrophobic acyl chain. So what I spent really, and I'm still doing it, 50 years looking at was, well, gee. Biological membranes, they have hundreds of different species of lipids. Well, why are they all there? And what are they doing? And some of them, if you put them in water, you extract them, put them in water, they don't adopt a bilayer structure, they'll adopt something completely different. So what roles do these non-bilayer lipids have? Similarly, they have asymmetric transbilayer distributions of lipids. The lipid composition on one side of a membrane is different than the lipid composition on the other side. And so ask the question, well, can we generate lipid asymmetry in response to some of the ion gradients you find across membranes? And what are the consequences of getting different lipid distributions? There's huge consequences, actually, but I'm not going to be able to go to that today. So what we discovered was we could use the NMR machine to study lipid polymorphism, or the different structures of lipids. You just take lipids, add water, and then they'll adopt various structures, say a bilayer phase. And we're looking at the phospholipids. They have a phosphorus in the head group. And they can rotate rapidly around their long axis. That gives rise to a characteristic line shape that we could then say, okay, the low field shoulder, high field peak, that was a bilayer line shape. And say, okay, then they have a cylindrical shape, if you want to think of it that way. These concepts might not seem to have much to do with vaccines, but actually turned out to be quite useful. Then other structures, such as the hexagonal phase, long tubes of water surrounded by lipid head groups. You get emotional averaging around those long tubes, and the low field peak, high field shoulder, that's diagnostic of the hexagonal phase. And a different molecular shape can be assigned to that. And so this was it could give us a great insight actually into, we could put a whole intact membrane in there, see the bilayer structure, et cetera. But in order to pull out what each role of what lipids are doing, you can't look at the intact membrane, it's just too complicated. And so we developed very simple model membrane systems, so little vesicles. We had to develop a machine to do this. This is called the extruder. Where we rammed bigger bilayer structures through small polycarbonate, 100 nanometer polycarbonate filters, and out the other end we get these unilamellar systems. This actually did kind of revolutionize things in terms of being able to study well-defined systems. Now, we then showed, we used these liposomes to show that what are these non-bilayer lipids there for? Well, if we put them in these bilayer structures, we suddenly found, well, they fused. They would fuse to form larger structures. So that's one of the reasons membrane fusion is kind of important, fertilization, interstellar transport, and all of that. And so you start to see a rationale or a reason why these other components are actually there. We also used these liposomes to demonstrate lipid asymmetry. So in order to do this, we constructed these small vesicles have a pH gradient. And we said, okay, well, what can we flip across the membrane to simulate lipid asymmetry? In other words, more lipid or particular lipids on one side versus the other. And we synthesized, this is let's talk about serendipity, but I'll say later. We synthesized this ionizable cationic lipid, which has the property that can exist in a protonated form or I'm going to emphasize this one. I don't know if this actually works, but no, it doesn't actually. But anyway, it has a property that, say, at low pH is protonated and positively charged, whereas at higher pH, it's deprotonated and net neutral. Well, the net neutral form can flip across the membrane very easily. If it's protonated and positively charged, lipid bilayers are remarkable, as they're highly impermeable to charged molecules. And so it'll get to the inside, get protonated in the low pH environments, about the acidity of a lemon, and stay there because it possibly couldn't get back out again. And so that was we did all sorts of things with that, which again I'm not going to get into, but we got distracted by the drug delivery potential. What we found was we could load cancer drugs into these vesicles to very high levels. This just shows pictures of these vesicles with doxorubicin inside. So we could get so much doxorubicin in there that it was past the solubility product and it crystallized out. So we have little nanocrystals of the doxorubicin on the inside. And so these, of course, this is a pretty good drug delivery system. And we're still working on this, actually. We got 2 drugs approved by the FDA and the EMA, and we're still working on ways to improve them further. I'm sure these are going to be really quite important in the fight against cancer. So we started a company that was back in 1992 to use these liposomes to deliver cancer drugs to tumors. So this is me and 4 postdocs in the lab. We had a good time with this company, but raising money in companies is kind of an imperative. And the CEO came to me one day and said, putting old drugs in liposomes, this doesn't work. We got to be doing gene therapy. So I said, okay. And we started off on that route. And so since then, we've spent about 27 years trying to develop systems to deliver siRNA and mRNA. So I'm going to go through three parts of this. First of all, the design of lipid nanoparticles. How do we encapsulate these negatively charged polymers in these nanoparticle systems? The Onpattro story. This is the first demonstration that these things could be actually useful for delivering RNA. And then I'll close very briefly on the vaccine story. So our aim in 1995 was to develop a delivery system to take a small interfering RNA, 13 kilodaltons, pretty big molecule in drug terms, to silence a gene in the liver. Now, this involves packaging it up in a lipid nanoparticle, and then we inject intravenously. It has to circulate around, go to the liver, which is where we were aiming to silence a gene, extravasate, get taken up by hepatocytes, we hope, get out of the endosome and actually do something. This is kind of a major challenge, should we say. And of course, the immune system is set up to avoid this happening at all costs. This is occasionally you start off on things where you're not quite sure what's going to happen. The first problem that we hit was that how do we encapsulate these nucleic acid polymers? They're obviously very negatively charged. Well, the only way to get them efficiently get them into a lipid nanoparticle is to have positively charged lipids. Well, nature doesn't like positively charged lipids. There's no positively charged lipids in nature. They're really, really toxic. You want to kill mice? This is a good way to do it. Well, how are we going to be able to use these lipids? How are we going to encapsulate? Well, this is where basic research comes in. So what we did was we tried DODAP, this lipid that we'd made to investigate lipid asymmetry. It had this property that low pH is protonated, positively charged. You can flip it across bilayers, but maybe we could also use it to encapsulate a nucleic acid. So in other words, at pH 4, it said the acidity of a lemon, maybe we could formulate it at that pH where it's positively charged, and perhaps things with the mRNA or sRNA, or in this case sRNA, would remain encapsulated. And we found it worked really well. We could incubate our RNA, DNA, we want to encapsulate in water at pH, buffered at pH 4, and then have the lipid in a ethanol solution and mix it together very rapidly. And one of the first things to fall out of the solution would be the oligo with the positively charged lipid, the ionized lipid associated with it. Do it rapidly enough, and the other components would then form what we term a limit-size vesicle. And so it turned out to be a very useful and relatively non-toxic, which was, of course, the primary thing, way of encapsulating nucleic acid polymers. So ideally encapsulating 100% trapping efficiencies. We can adjust the size by changing the surface lipid to core lipid ratio. It's scalable, reproducible. I should say we did this before we really went into animal experiments because we knew we couldn't use the cationic lipids. So now I'll move into the first application, which is the Onpattro story. This is to treat a drug that's already been, or treat a disease that's already been mentioned called transthyretin-induced amyloidosis. And so this is a protein that's made in the liver. So I'll talk a little bit about that progression. So after a conference I attended, this was in 2004 in London, I put here, I was pursued. Victor, I often characterize him as a mad Russian. He has those characteristics. So somewhere in Soho, where we ended up, he was saying, well, we have a delivery problem. How do we get our small interfering RNA into hepatocytes in vivo? Because Alnylam was founded solely to use small interfering RNA as a therapeutic to silence particular genes in whatever tissue to inhibit the production of pathological proteins. So they had a problem. They had to. This resulted in a collaboration with Alnylam that went on between 2005 and 2012. That involved INEX and my lab as well as Alnylam. We had teams in both places, and we synthesized many different formulations over those years. We started with the question that, are these things any good? We can get them into a lipid nanoparticle. They aren't too toxic, but do they do anything? with the ionizable lipid there. Well, lipid nanoparticles get into cells by endocytosis, has already been mentioned, and we want to design them so if they basically deliver the contents to the cytoplasm as opposed to the lysosome. We need to destabilize the endosome is the bottom line on this. Well, how do we do that? Well, this is again where basic research turned out to be useful, because what we found was that if the anionic lipids or negatively charged lipids that you find in biological membranes, if we had a positively charged lipid to that, it flips it straight over to these non-bilateral phases. In other words, become very, very destabilizing for membranes. And so this gave us a kind of design approach to go after the ionizable lipids, that at some point they had become positively charged and then destabilize the endosomal membrane. So that's the way, this was a design feature that we followed during this, well, we're still following it today, but during that collaboration with Alnylam for sure. So engendering the maximized potential for formation of non-bilateral structures when they become positively charged. Now we assessed the in vivo potency of these systems using a factor 7 model. So this factor 7 is made in hepatocytes and obviously goes into the circuit clotting protein. So if we're able to silence factor 7, then say 24 hours later, there's less factor 7 in the circulation. So it was a reasonably high throughput model to test our systems, test the efficacy of the systems. Now, many different ionizable lipids were formulated, but what we found was that the potency was enormously dependent on the pKa, in other words, the pH at which these lipids got protonated. As the pH in the endosome goes down, as the endosome matures, it goes from pH 7.4 down to about 5 or less. And so obviously these things are sensitive to that environment. Now this was an amazing result actually. We found that the potency was remarkably on the pKa. This is a log plot that you're seeing here of the potency 1 over the effective dose where we see a biological effect. And if you moved as little as, say, 0.5 units away from an optimum pKa of about 6.4, the efficacy was reduced by 100-fold or more. So very, very highly dependent. And over the course of the collaboration, we went from about 10 mg per kg, where we could see some effects of gene silencing, down to 5 micrograms, without increasing the toxicity. And so this is where the therapeutic index of 1,000 came from. So, okay, well, this is where the clinicians at Alnylam said, okay, well, I think we got something that's ready for the clinic. What should we go after? And they chose to go after transthyretin-induced amyloidosis. This is a remarkable statement here, actually. I mean, because we can silence factor 7, that means we can silence any gene in hepatocytes. So just with the same form, you switch, you go use a different siRNA, and then you can go after a different disorder. One of the charms, it's an amazing, these genetic approaches are so powerful because they're so general. Anyway, hATTR amyloidosis, so transthyretin is a tetrameric protein. If there's mutations, it can form these fibrils, which presumably deposit everywhere in the body. They have really nasty effects on nervous tissue and cardiac tissue. And so heart failure, et cetera. About 50,000 patients worldwide, there's no effective therapy, usually fatal within 5 years. This picture has already been shown. It's a really nasty disorder when you think about it, because if you have trouble walking in your 30s, and you have this disorder, you're very likely to know parents, grandparents, uncle, whatever, because it's a hereditary disorder. You'll know what you're in for. So this is not a pleasant prospect. So a potentially very simple solution here, just silence that gene. Let's stop that protein being made. And so this is designing an siRNA to silence transthyretin, and then you're going to reduce the levels of transthyretin in the blood. Maybe if you get very lucky, you'll get some regression as the amyloid fibrils are dissolved. So really potentially simple. This is the design of these medicines is much simpler than small molecules, that's for sure. So the phase 3 study was performed by Alnylam. So this was done, the results were announced in 2017. 148 people with administered the transthyretin drug, the siRNA to silence transthyretin, or sterosaline as a placebo. Primary endpoint was the neural impairment score over an 18-month period. Secondary endpoints, the quality of self-reported quality of life, ability to walk, body mass, obviously a wasting disorder, etc. Now, this was where, over this 18 months, what was found, if the individuals on the placebo, the neural impairment score got steadily worse. So they were not doing well. But remarkably, the people in which we were silencing the gene in the liver, if anything, they got better. Now, this is people that are already established and progressing hereditary disorder, and here we are actually impacting that in a fundamental way. These are the best clinical trial results I've ever seen. They were announced in 2017. I always characterize the p-value for the improvement, the neural impairment score endpoint, as there's no doubt. This is 1 over Avogadro's number. I mean, there's absolutely no doubt that this drug works. And similarly for the other endpoints, quality of life, ability to walk, and so on. So potentially, really a curative therapy for what was previously a fatal disease. That's kind of remarkable. I thought this was the high point of my career, as you can imagine. Anyway, more was to follow. So this was approved. This drug was approved in 2018 by the FDA, and it's been approved in many other jurisdictions. A pretty big deal, clinical validation of the lipid nanoparticle approach, but also dramatically demonstrates the power of these gene therapies. So I'm going to go on now for the last few minutes just to describe what happened with regard to the vaccine application. In 2012, when Onpattro went into the clinic, as scientists, you're not going to change a formulation when something is in the clinic. Otherwise, you got to start all over again. So what were we going to do? And so I said, okay, we're delivering 13 kilodalton and siRNA. Maybe we can deliver a messenger RNA, and maybe we can get that to express a protein in the liver, as opposed to silencing a protein. Maybe we can get hepatocytes to make a protein that we want to express. So this is kind of, we have 1,000 copies of siRNA in a lipid nanoparticle. With a, say, 2,000 base mRNA, maybe we have 2 or 3 copies, where it's that much bigger. Anyway, so we wanted to find out whether this was possible, and it turned out to be remarkably possible. And so what we found is that these systems we can use the liver as a bioreactor to produce any protein. This is a heck of a statement. Child is born lacking a certain protein. Well, gee, let's get their liver to make it. Your liver makes 70% of the proteins in your body. Seems like adding a few more, it's only too happy to do it. So it's a revolutionary approach when you think about it. This is in pigs. If you want to win the Tour de France, we got a solution for you. Expression of erythropoietin, super physiological levels. So but this is where serendipity came in again. We were approached in 2014 by Drew and Katie. Needed a delivery system for mRNA vaccines. And as you've heard, how they managed to beat some very fundamental problems in terms of the immunogenicity of these systems and the levels of expression, but they still had a delivery problem. They still had to be able to get these big molecules into target cells to get the kind of responses that you've already heard about here, MHC1, MHC2, et cetera, in antigen-presenting cells and muscle cells. And as you've also heard, this was a paper of the year in 27. Anyway, the Zika virus vaccine, really quite remarkable. Now, all we're doing here, as you saw, we had the erythropoietin or marker protein or whatever we were expressing in the liver. All that was done here is take pretty much that same system and then encapsulate a messenger RNA that coded for the Zika virus pre-membrane and envelope glycoprotein. And so this was then injected intradermally, and then in mice, and then challenge, 2 weeks, 20 weeks later, and complete protection against infection by the Zika virus. So this is as you've already heard, but pretty amazing that you could just take these systems. We were optimizing for the liver, but boy, suddenly they started to work as vaccines. So you got to get lucky in life. And this was definitely. There's some other things that happened, so this made up for a lot. Anyway, so in 2018 or so, Acuitas, this was a company we co-founded, began working. This is on the basis of the Zika virus and other results. Began working with BioNTech to develop influenza vaccines, and BioNTech was also working with Pfizer on a flu vaccine. So. Obviously, in January, February 2020, all efforts, went into making a COVID-19 vaccine for obvious reasons. And so here it's just again a matter of swapping out the mRNA for an mRNA coding for the SARS-CoV-2 spike glycoprotein. And you all know the results here. That in November of 2020. The press release, I remember reading this press release, I couldn't quite believe it. 95% effective for preventing COVID-19 and across all age and gender, race, et cetera, reasonably well tolerated. At the bottom you'll see they were anticipating perhaps making a 1.3 billion, it's a big number, doses by the end. I think they're closer to 3 billion by the time it actually finished. So approved by the FDA and many other regulatory agencies, and has played a major role in containing the COVID-19 pandemic. So it's been a pretty amazing journey. And what I hope I pointed out is that a lot of that depended on very basic research and having the tools and the insight that gave us to be able to design these lipid nanoparticle systems for that had application as vaccines. As also been pointed out, particularly by Drew, the journey is just beginning. This is really, I always term it the third generation of pharmaceuticals, first generation small molecule drugs, second generation biologics. But the gene therapies, protein replacement, vaccines, gene editing, it's just opened up a tremendous variety of new potential therapeutics. And you're going to see these really becoming fairly dominant over the next, I think within 5 to 10 years, we're going to see people are projecting as many as 40% of new drugs are going to be based on this approach. The charm, one of the amazing things is how quickly, and it's already been pointed out, but once you know the protein you want to silence or express or edit, then you can make the siRNA, mRNA, et cetera, within a matter of a month or two, encapsulated in a day, and you've got this highly targeted personalized therapeutic available. So we're going to see many, many new drugs, as I mentioned. I got to close by acknowledging this is the work of hundreds, if not thousands, of people that I've really talked about here. But one of the things that we've done in Vancouver is keep a team together. That team, the group that I showed that where we formed Inex Pharmaceuticals, we still work together 40 years later. So like Nick Hope, Tom Madden, and many others, Steve Ansel, Ying Tam, Barb Mui, and Paulo Lin, 20 years or more. The Al-Nilam team, as is indicated here, had a huge influence. Mark Tracy at Kinnekink and Martin Mayer, Manu Maharan. Marcus Siflini in UBC Chemistry Department was the guy that really broke the problem of the ionizable lipids, where we just said, how do we get things that are more toxic or less toxic? Turned out he made one that was not just less toxic, but also more efficacious. These things work in their brain, but I haven't talked about that. My own group, and of course Drew at the University of Pennsylvania, has played a huge role here. So with that, I'll close. Thank you very much.
Incredible, fantastic. Thank you, Dr. Cullis. We will now hear from our next early career investigator, Dr. Isabel Malhame, Assistant Professor at McGill University, Obstetric Medicine and General Internal Medicine at McGill University Health Center, and FRQS Junior 1 Scientist at the Research Institute of the McGill University Health Center. Dr. Malhame was selected by Zulfikar Bhutta for her research on reducing severe maternal morbidity across the pregnancy continuum. Delighted to hand it over now to Malhame.
Thank you so much. Thank you so much for this opportunity to present our work and future progress on research to reduce severe maternal morbidity. And particularly thank you to Dr. Zulfikar Bhutta, who has dedicated his entire career to improving the health and well-being of children and mothers globally, and who has made sure through his advocacy and incredible legacy that evidence-based medicine did not leave them behind. I would also like to acknowledge the fact that the land where I conduct my work, Tiohtià:ke, is the traditional territory of the Kanien'kehá:ka, a place which has long served as a site of meeting and exchange among nations, and that continues to carry this tradition of cultural mixity forward. I have no conflicts of interest. Severe maternal morbidity is a set of unexpected outcomes related to pregnancy, labor, childbirth, and the postpartum period, resulting in severe illness, prolonged hospitalization, or long-term disability. Severe maternal morbidity is on the pathway to maternal mortality, and as a result, for every woman who dies during or after pregnancy in high-income settings, about 85 of them experience a severe maternal morbidity event. The World Health Organization has thus recommended a focus on maternal morbidity to monitor and improve the quality of obstetric care. Unfortunately, in Canada, the incidence of severe maternal morbidity has been on the rise. While certain types of severe maternal morbidity events, such as serious infections or postpartum hemorrhage or surgical complications, have been improving, on the contrary, other medical complications, such as assisted ventilation for respiratory distress, acute renal failure, or stroke, have been increasing. And this may be explained by transitions in the obstetric population, which now includes women who are at higher risk of complications during pregnancy and postpartum. Women are getting pregnant at an older age. The prevalence of obesity has been on the rise. Women are developing more hypertensive disorders before, during, and after pregnancy, and more metabolic complications such as diabetes. As a result, we're now seeing a paradigm shift in causes of maternal mortality in high-income settings, which now comprise cardiovascular disease and other medical disorders. So how do we, going forward, reduce this deterioration of health from morbidity to severe maternal morbidity and prevent this escalation of care? And this is the research question that my program has been trying to address along three major axes, the first being risk prediction, to identify individuals at high risk before they develop adverse complications. But first, before prediction, it is important to define the outcome that we're interested in predicting. And we put forth the concept of cardiovascular severe maternal morbidity, a composite outcome regrouping cardiovascular indicators of morbidity such as acute myocardial infarction, pulmonary edema, and stroke. And we've operationalized that definition into an algorithm of diagnostic codes that can be used in administrative data sets to do more research on that topic moving forward. And we then characterized this outcome of interest and looked at a broad, large population sample and saw that temporal trends over the past years were revealing that cardiovascular severe maternal morbidity was in fact increasing, and that most maternal cardiovascular deaths were in fact occurring in women who were not known for cardiovascular disease prior to pregnancy. We were then interested in predicting cardiovascular adverse events in the large population sample that comprises the general obstetric population, and not just women who are known for cardiovascular disease. We developed two risk prediction models, one to predict adverse events occurring at delivery, and another one to predict adverse cardiovascular events occurring postpartum. And both risk prediction models performed extremely well with good accuracy and good internal validity. What also came to be apparent was that preeclampsia and severe preeclampsia were among the strongest markers and the strongest predictors of cardiovascular severe maternal morbidity. So we were interested in developing a risk prediction tool specifically for this subset of the pregnant population. And while clinical risk factors alone allowed us to have a prediction model that had a reasonably good accuracy, we are now interested in future years in implementing risk prediction tools that incorporate biomarkers, including echocardiographic and circulating biomarkers, to predict cardiovascular morbidity in pregnant women with preeclampsia. The second axis of my research program centers around the management of high-risk condition to help develop better early diagnosis and more tailored therapies to women with conditions at risk of adverse events. B-type natriuretic peptides have long been used in the non-pregnant setting to diagnose heart failure, but we were interested in assessing whether this diagnostic tool could be used in the pregnant population, and found that in fact it was quite sensitive and specific and helped us greatly to make the diagnosis of heart failure in the pregnant population. And women with heart failure in pregnancy represent a heterogeneous group of patients. Women with peripartum cardiomyopathy, a pregnancy-specific type of heart failure, who also develop preeclampsia, are actually at higher risk of adverse events, including pulmonary edema and thromboembolic complication, and as such may require a more tailored therapy. And finally, to make sure that pregnant and postpartum women have the best therapies available to them, it is important to advocate for their inclusion and their participation in clinical trials. And during the COVID-19 pandemic, we highlighted that clinicians who look after pregnant women with COVID-19 would actually be strongly in favor of including them in clinical trials of intervention for COVID-19. And we hope that this advocacy can also be translated in other non-obstetrical conditions. And in the next future years, I will focus on atrial fibrillation in pregnancy, which is a type of arrhythmia that can be associated with severe morbidity and mortality that is poorly understood. Now, the third axis of my research program focuses on quality improvement and patient safety. And so in order to reduce severe maternal morbidity events, it is important to learn from one another and decrease the adverse events that occur. And to do so, we are interested in developing an infrastructure to review in a synergistic and centralized manner severe maternal morbidity events across the country. An obstetric survey system is a nationwide, centralized, anonymized case reporting system for severe maternal morbidity events. An obstetric survey system would be particularly well suited to the Canadian context, as it allows us to engage with maternity units within communities, to respect regional specificities and priorities, to conduct an in-depth appraisal of individual severe maternal morbidity events, including a holistic assessment of clinical and social determinants of health. Our team is currently in the process of mapping out maternity units across the country and identifying local severe maternal morbidity review systems in place. We have so far contacted 230 maternity units, and we're collecting answers to our survey. And we so far have found that the majority of respondents do have a severe maternal morbidity review system in place that could be leveraged in an obstetric survey system, as they would be willing to share anonymized data about adverse events occurring in their unit. So in future years, we will be working to develop and implement a Canadian obstetric survey system called CANOS to reduce severe maternal morbidity moving forward. In conclusion, to reduce severe maternal morbidity, it is important to refine and validate risk prediction tools, to generate the evidence base to manage high-risk conditions, and to develop the infrastructure to learn from one another and reduce adverse events. And with that, I would like to acknowledge the broad network of collaborators and co-investigators who have participated in the work and the results that I've presented, as well as the supporting organizations and the funders that have supported our work. I would also like to thank my mentors, including my senior mentors and peer mentors, who have made this early career journey that much more richer and enjoyable. Congratulations to all the Gairdner Laureates. You are an inspiration to us all.
Fantastic. Thank you so much, Dr. Malhameh. I am now delighted to introduce Dr. Zulfikar Bhutta. Dr. Bhutta was awarded the 2022 John Dirks Canada Gairdner Global Health Award for the development and evaluation of evidence-based interventions in child and maternal health for marginalized populations, focusing on the outcomes for the first 1,000 days of life. He is the Robert Harding Chair in Global Child Health, co-director of Sick Kids Center for Global Child Health, senior scientist at the Hospital for Sick Children, professor in multiple departments at the University of Toronto, founding director of the Center for Excellence in Women and Child Health and Institute for Global Health and Development at the Aga Khan University, South Central Asia, East Africa, United Kingdom. Delighted to turn it over to Professor Bhutta.
Thank you very much, ladies and gentlemen, and thank you for that generous introduction. Let me get this technology sorted out. So I'm going to, first of all, it's been a thrilling morning of science. And what I'm going to share with you this morning is science of a different kind, implementation research at scale, which can influence policy, particularly focusing on some of the highest global burden countries affecting women and children, and also something that relates to global targets and goals. So I have no conflicts of interest to declare beyond a profound interest in the health of women and children across the world. And I'd like to start by first acknowledging that although I'm very happy to be recognized as an individual, in global health, and particularly global public health, individuals count for nothing. Most of the work that we do is done in teams and in collaborations. So I want to start by acknowledging the very large team that I work with in Pakistan and in East Africa. And many of the people that you see here are the real heroes, and several have worked with me for over 2 decades. And I have another similar team, relatively shorter, but now also spanning over a decade here in Toronto, of young graduate students, scientists, and collaborators who have similarly worked on these problems. So I want to take you back over 2 decades ago, when some of us assembled to look at the state of child health across the world. This was around the beginning of the Millennium Development Goals, with not a lot of information, and one of the issues that we tackled was how many children were dying every year under 5, and where. Each one of these dots on this global map represents a cluster of 10,000 deaths. And when we drew this map in Bellagio, we recognized that a lot of this mortality was clustered in sub-Saharan Africa and South Asia. And South Asia appears as a red blob here, largely because of population density. But very little was known beyond the fact that these deaths were happening. One of the first questions was, do we know something about when these deaths were taking place? Because early childhood, 0 to 59 months, is still 5 years. And we set up to ask ourselves a series of questions around how this related to the entire journey of pre-pregnancy, pregnancy, childbirth, and the immediate post-birth period. So if you look at risk of mortality, it's no big surprise that most maternal deaths cluster in and around childbirth, as do some things that we don't talk about, stillbirths. The babies were born dead, early newborn deaths, and then child deaths. And you can well imagine that if you took this continuum, the first 1,000 days, where I've focused a lot of my work over the last 3 decades, represents that area of maximum risk. Pregnancy, first 2 years of life. And if you were to drill down even more on where these deaths were taking place, the very first day of life, which is supposed to be one of the happiest days in the lives of many mothers and families and parents, is also the saddest day in the life of many. Because close to around half of all stillbirths, about 30 to 40% of all newborn deaths, and a large proportion of maternal deaths occur in that early 24-hour period. At the time when we looked at this was really not on the global radar screen as a target or as an indicator that people would follow in countries in terms of addressing interventions. The second big question was, where were these deaths taking place? And to ask that question, we again had to go back to the basics and look at the structure of health systems. And in many places like South Asia, the health systems did consist of brick and mortar facilities, basic health units in villages, in rural areas, mid-level centers, tertiary hospitals, or district referral hospitals. But the quality of care in many of those facilities for particularly serious problems of mothers and children was generally so variable and poor, but when it came to a choice, people voted with their feet. They did one of two things. They either tried to find their way up the upper end of the health system, traveled hundreds of kilometers to a big city, or they stayed where they were. And that's why in survey after survey that my team was involved in South Asia and in East Africa, we found that 50 to 60% of all newborn and infant deaths were in village or rural settings. About a third were occurring in facilities, and a few, sadly, died on their way. Now, if you remember nothing from today, but you remember this infographic from 20 years ago, it will help you understand how some of the strategies that we deployed at that time, through rigorous trials, were focused on addressing this major gap in terms of access and care. And this gap also reflected the challenge that the world faced in terms of the distribution of the health workforce. I was part of the Joint Learning Initiative when we drew this map many, many years ago of how the areas with the highest mortality, sub-Saharan Africa and Asia, were also the regions of the world with the least distribution of qualified health workforce. So the first big challenge that confronted us was beyond just the trickle-down effect of development, which would take Africa 100 years and Asia probably 50 years to reach the levels of development of high-income countries. What could you do in the medium to short term to address this care gap? So could, for example, we deploy, through task sharing, community health workers to make a difference? And remember, we were interested in task sharing, not task shifting. So to do that, one would look at several models using the same health system that I've just mentioned to you of working directly with families and communities, or through outreach workers, community health workers who are deployed from health facilities, and packages that you could administer in facility care. Now, I've spent the last 25 years or so doing large cluster randomized trials, and I'm going to try and summarize some of this information in a few examples. Each one of these trials, just to give you a sense, takes about 5 to 6 years to implement, to complete from inception to completion, between $5 to $7 million on an average for each cluster randomized trial. These are effectiveness trials in real health systems with real public sector health workers. And more often than not, you wait 7 years and you sometimes get a negative answer. So to the students in the audience and to my own team, I often tell you that I have learned more from the negative trials that I've done. And I believe the world has learned more from the negative trials in terms of how not to do things than you do from the few when you are serendipitously fortunate enough to get results. So the first question was, could we work with public sector, not people that you can hire and fire, but government health workers in these community settings to reduce perinatal mortality? And could you do this through community engagement? And the program that we picked to work with initially was the Lady Health Workers Program of the government of Pakistan, which is largely assembled to work on family planning and primary health care, but did not have a focus on perinatal care or newborn care. And in this initial cluster randomized trial, we looked at a population level in rural Sindh and did two things. One, we created these village committees through mobilization and encouragement, which were basically gatekeepers, but men who supported these female health workers and gave them access and allowed them to work with communities. And then the lady health workers did not need a lot of support beyond encouragement to get women together and to do a positive deviance set of examples of how could they make their want better, how could they work towards improving care during pregnancy and during childbirth. We had very little hopes that we would get anything out of this because most of the studies to date had been small-scale efficacy trials. And what we found after 4 years was that some of the results were phenomenal. First of all, very clear differences in the intervention villages versus control villages in terms of antenatal care attendance, care in and around childbirth. People went and bought clean delivery kits. We didn't provide them. They were able to understand the value of a clean birth, looking at even practices that were culturally difficult, like delaying bathing the baby, which would lead to hypothermia or, let's say, an increased risk exposure. And importantly, an impact on hard outcomes. So when I got these first results, as I often tell my students, of an impact on stillbirths, I told the team to go back to the drawing board because that was impossible. We had not put a single obstetrician in the health system. And to have an impact on stillbirth required that you had something at childbirth. For example, increased cesarean sections. And we hadn't done that. So we saw the impact on neonatal mortality. Stillbirths were difficult to imagine. They went back, came back and said, no, the result is there. And then the penny dropped that what had happened at a population level over time, over these 3 years, was a very consistent and a steady increase in care seeking within the same health system that we had not really put any more physicians in. This was one of the first trials at effectiveness level of the role of community health workers and very rapidly became global policy, with also India adopting this through its ASHA workers. The next big question was, could we train these health workers to do a little more? And one of the big challenges at that time was neonatal infections, and particularly neonatal infections of the kind that related to cord care. To students in the audience, omphalitis is not only very common in about 1/5 of all births, but it can kill, particularly if it is associated with serious pathogens such as Staphylococcus aureus. So the question that we tried to address was, could we do something about this at domiciliary level, at home level, particularly for births that are taking place at home? And in this cluster randomized trial, the design was a factorial design, but very simple, set to answer 2 questions. Could we provide some soap and education to encourage families to wash their hands? And could we include within a birth kit something simple like chlorhexidine to encourage families to clean the cord with and not apply all other kinds of gunk that typically happens in these rural settings? And the district that we chose was Dadu, which is one of the poorest districts of rural Sindh. And as you can see, the intervention isn't rocket science. We provided the chlorhexidine in a small bottle with some instructions to apply it every day till the cord dropped off. And the results were extremely educational. The first important thing to learn was that handing soap and education around hand washing bombed, didn't work. And in hindsight, we could have predicted that because this was a water insecure area. When people barely have water to drink, it is very difficult to convince them to spend about a liter of water every day washing your hands, and repeatedly. So despite all the exhortation, that didn't seem to make any difference. But chlorhexidine had a massive impact in terms of both infections and also in terms of neonatal mortality. This 40% reduction almost in neonatal mortality was remarkable and quite extraordinary. And it needed to be confirmed in other contexts and geographies at scale. And that's exactly what happened. That in South Asia, in Bangladesh, and in Nepal, similar studies, some with a slightly different design of not daily application, but single application. And you can see consistently in this Kaplan-Meier curve that you could see very clearly that it had a consistent effect. And this very rapidly became global policy at the World Health Organization in terms of what to do about newborn care in facilities and also in community settings where births were taking place in less hygienic circumstances. In our ambition to do more with community health workers, we also tried to look at another killer, which was pneumonia in young infants, and particularly severe pneumonia, where the only alternative was to send them to a hospital and to provide them hospitalization and potentially intravenous antibiotics. And pneumonia, to many in the audience, is a killer, particularly in young children who are unimmunized. Today, we are dealing with a fortunate situation that globally pneumonia deaths have gone down because of the widespread use of hip vaccine and pneumococcal vaccine. But I'm talking about 20 years ago, when this was really not at scale at that time. And the only recourse was early diagnosis and management. So in this cluster randomized trial with community health workers, that's exactly the question that we asked. Could they recognize severe pneumonia early? Could they treat it? But to do this trial, we had to set up an additional safeguard of somebody making sure that kids did not drop out of the safety net and had an adverse outcome. So our endpoints for the study were more like identification of treatment failure. And in this large cluster randomized trial, you can see that we proved the point that you could train community health workers in recognizing, managing pneumonia. And overall case fatality rates in both arms were less than 0.5%. So I was very happy, at least, that the point had been made. This became global policy transiently for about 3 years before vaccines kicked in. But at the time when it did, it made a huge amount of difference for places which had no access to pneumococcal vaccines and others. But this is all about deaths. And deaths are a sad, hard endpoint. But there is much more to death. And as you saw from the preceding presentation, morbidity and adverse outcomes also relate to things that do not necessarily lead to mortality. So our interest shifted over this period of time to also looking at things beyond mortality. And a large part of my group's work shifted to looking at nutrition. And as some of you might know, one of the biggest global challenges that we have is linear growth retardation. These curves of how children grow across the world tells you how we live in different worlds altogether. These are length for age z-scores, and as you can see, in many regions of the world, the Southeast Asia, which is largely driven by South Asia and African region, children deviate very quickly after birth. And by 6 months of age, by 24 months of age, a large proportion are stunted, which means that they are below their expected height for age. You can see that in this group of African children. The line that you see in chalk on the wall is what you would expect to be the average height of children at that point in time, looking at global standards. You would never notice in that community that anybody was abnormal or different, because the normal here is what you see. And that's one of the reasons why in many global contexts, governments miss the point that 40, 50% of children under 5 were stunted. The numbers were huge. And even today, as we speak, there are close to around 150 million children worldwide who are stunted under 5 years of age. And most of this occurs within the first 24 months of life. With my friend Chuck Nelson, who identified how important this period is in terms of not just growth, mortality, but also development. And if you look at how many things such as language, sensory motor functions, and higher cognitive functions develop over time, you can see how important this first 1,000 days, the first 2 years of life and the period of pregnancy is in terms of optimizing neurodevelopment. Some of these impacts on these children are not just short-term, around the time when they live. And if you were to look at across the life course, some of these adverse early exposures, particularly in the first year or two of life, can have lifelong consequences, which we are beginning to recognize now. They're not just limited to that period. If COVID has taught us one thing, is that how important the school age period is also in terms of adverse consequences that impact children well into adolescence and adult life. So we didn't know a lot of that at the time when we designed these interventions. A simple question that we wanted to answer was, can you do something about this at scale in population settings? And with my colleague Aisha Zousafzai, we designed a cluster randomized trial to try and answer that question. And this was done with community health workers working in rural Sindh, different area, where we tried to see if you could integrate early child stimulation with early child nutrition, education, and messaging. It took a little bit of time to develop this. The cluster randomized trial, again, large scale, hundreds of children and different villages. And the intervention was based on what was available at that time from UNICEF and others as a stimulation package. This has improved since, but it was basic around interventions that parents can do. We all do intuitively, like singing, talking to the child, eye contact, stimulation. The results were very interesting. We were able to demonstrate a huge impact in terms of developmental outcomes by 2 years of age. This has subsequently been shown to be true at 5 years of age as well. The tools were limited, but using Bailey's, we were able to show that. And we also found that it did very little for nutrition. If you looked at linear growth, despite the fact that these children were stimulated, education by itself in this food insecure population did very little. So the next big question was, could we improve nutrition? But WHO picked this up, and these trials, particularly this cluster randomized trial with community health workers, was the basis for the development of the nurturing care framework by the World Health Organization and UNICEF. And this framework is based on 5 indivisible essential components. And they include health, safety, nutrition, environment, and protection. And these are extremely important in terms of providing children the safety nets and the safe platform on which to layer everything else. In this period of time between the middle of the MDGs and the end of the MDGs, this was an exhilarating period. And both with my team here at Sick Kids and in Pakistan, we were able to influence several large Lancet series based on collation of the evidence, synthesis into practical, easy to implement, understandable packages of interventions that governments could pick up. The Lancet 10, these nutrition interventions that we developed in 2011-12, were taken up by many government planning bodies and bilaterals as the platform for scaling up nutrition interventions. First big question that they asked was, how much difference would it make? And we were able to show that in a large number of countries which had close to around 80-85% of the stunting burden, that these could make a lot of difference, both individually and collectively. We were also able to cost it at that time ourselves in terms of cost per life year saved or DALYs averted. And the World Bank picked it up in terms of developing a costing model. This costed model for the global framework for nutrition asked for $70 billion invested over 10 years, which, if you look at how much we have spent on wars and everything else since, is nothing. This is what would it have taken. the world to implement some of these strategies. But because advocacy and science-based synthesis of evidence itself is not enough, tremendous pressure upon us to, okay, walk the talk, show us that it can be done. And it can be done at population level, at scale, and it'll make a difference. So even though I had told our team that I was truly not interested in doing a trial around the first 1,000 days interventions because they were all based on solid science, and it made very little sense to go and do it, we were forced to implement this in the field to demonstrate that it can be done, and to do it with government health workers. So in our most recent cluster randomized trial, asking that simple question, do these nutrition interventions in the first 1,000 days make a difference? We've had to do exactly that. And this was done in, again, another district of Sindh. And I showed this particular slide to various audiences to make the point that this was an effectiveness real-life trial. And that's why the coverage of what we got is around 50–60%, which is what you get in real-life setting. In an efficacy trial, we would have got this coverage up to 90% or thereabouts. But because this was people making a choice, whether they took a commodity in pregnancy, whether they took a commodity during breastfeeding, or they implemented something after birth, we were dependent upon what people would do in real life. So compliance, 50–60%. Did it make a difference? It made a huge difference from interventions in pregnancy, both in terms of low birth weight, in terms of wasting in the first month of life. We saw a consistent difference in terms of reduction in stunting in these children right up to 24 months of age. And importantly, something that's very resilient to change is a reduction in wasting. Others wanted to see more. A cluster randomized trial is at best at a population level of about 1 million. They wanted this to be at large national level. And to do this, our team at SickKids had to do what we call exemplars, is to look at the best performing countries in real-world setting to see how had they improved nutrition. And these exemplar studies that have been published looked at the 5 best performing countries in terms of how nutrition changes, how stunting reduces. And it made a very important observation that in addition to the kind of commodities and nutrition-specific interventions that I've been talking about, that you also needed to invest in contextual interventions, to improve women's education, to invest in family planning, to increase women's empowerment, and to reduce poverty. So those conditionalities were just as important as giving people iron folic acid tablets or improving things after birth. And this has now been shown convincingly across a much larger number of countries. This was published in The Lancet last year, and through examples which are at subnational level, that you need a combination of both. So in terms of going forwards, that's the policy imperative of not just restricting interventions in nutrition to the health and nutrition sector, but to go beyond that. So as I close, what does the next decade or two look like? So the first important thing to the students in this audience is, despite the success that we have had in the Millennium Goals, we still have an unfinished agenda of reducing premature or preventable mortality. We just have a wider age band. In the MDGs, we were focused on children under 5. But if you take the extension of children from 0 to 20 years of age to adolescents, we still have close to around 9 million preventable deaths, particularly in children who have been ignored so far, the school-age children and adolescents. So we have a mountain to climb still, beyond the mountain that we have already climbed. The second is that you need to extend the nurturing care framework to beyond the first 5 years of life. So the principles of protection, nurturing, care, safety, optimizing health and nutrition is just as important for school-age children and adolescents as it is for young children. Now, this is something that as parents we know intuitively, but in global policy has just not been the framework that has guided our work. So I'd encourage students in the audience to read the most recent Lancet series that we published earlier on this year on optimizing child health and development, right up to adolescence, that I hope will guide future policy. So let me finish by saying that the Sustainable Development Goals are here. We talked about those yesterday. These are goals that most of us are not going to be able to reach because they finish by 2030. But already we're talking about how can we go beyond the 2030 targets. And my premise to you is that women and children, particularly young children, need to be at the center of the Sustainable Development Goals for us to reach our human capital potential. Thank you very much for your attention.
Please join me in thanking all of this morning's speakers for those insightful presentations. We're now going to have a very short bio break, so please feel free to grab some refreshments, stretch, and we'll be back promptly at 11:40 a.m. Thank you.
Hello, everybody. We should perhaps get started for the next few sessions, so I'm encouraging everybody to sit down and if those of you at the back could make your way to your seats, that would be much appreciated. Thank you. Hello, everyone. I want to welcome you back to these sessions. It's my privilege to be here today. My name is Sunny Coley. I am the head of medical and regulatory affairs at Roche Canada, and I'm a practicing physician in internal medicine and ICU at Oakville Hospital and adjunct faculty at McMaster. So it's a privilege for me to be involved in this today. I'm happy to join you as chair for the second portion of this event. At Roche, we are passionate about advancing science, no matter where it comes from. Only by partnering with the brightest minds can we serve patients with difficult to treat diseases and make sure that we deliver the right innovation to the right patient at the right time. And that mandate certainly resonates with me. In my previous role, I've been in this auditorium a lot. I was the co-founder of a digital health startup company that was actually supported by Mars. So I lived here and I lived in this auditorium, so it's weird to be here in another role. So innovation is a mandate it's really near and dear to my heart. So that is why I'm honored to introduce the leading researchers of our time who are pushing the boundaries of biomedical science. So with that in mind, our next early career investigator speaker is Dr. Anna Maria Kopescu. She is an allergy, immunology, and internal medicine specialist at the rank of assistant professor at McGill and the University Health Centre of Montreal. She was selected by Dr. Deborah Cook as part of a team leading a multi-center randomized control trial on point-of-care penicillin allergy testing, which is near and dear to my heart as an ICU doctor and an internist. So I'd like to welcome Dr. Kopescu to the stage.
So thank you very much for that lovely introduction. I see people still coming in. I will wait just a couple of seconds, but I'm going to start. So my aim is really to convince you that by improving patient care, we can also focus on penicillin allergy. So first of all, I wanted to thank very much Dr. Cook for the opportunity to present today. I also wanted to thank the foundation very much. As you all know, Dr. Cook is a distinguished professor at the McMaster University in critical care medicine. By designing and conducting landmark national and international studies, her work has helped decrease the important human and economic costs of critical care illness. Her work has a direct impact on patients, their families, the healthcare systems, and societies. Her passion for creating measurable benefits for our patients is truly an inspiration for our particular research. So why is our research critical? It has been estimated that by 2050, 10 million worldwide deaths could result from antimicrobial resistance, making it deadlier than cancer. So let's try to understand how antibiotic resistance is developed and focus on antibiotic allergy and particularly penicillin allergy. Indeed, 10% of the Canadian population will report a penicillin allergy. However, it is now known that less than 1% are truly allergic. Please also note that when we focus on the severe cutaneous adverse drug reactions that have important mortality and morbidity, these have been reported in about 1 in 1,000 patients. So when a patient actually has a reported allergy or a true allergy to penicillin, this will lead to the prescription of broad-spectrum alternative antibiotics. The use of these antibiotics has been unfortunately associated with an increased healthcare cost, increased risk for antimicrobial resistance, suboptimal antibiotic therapy, medication error. Using these broad-spectrum antibiotics leads to undesirable side effects and also risk of hospital complications. It is needless to say that this antibiotic allergy is a national and international public health concern. And in this context, there has been increased focus on penicillin allergy and its impact on antimicrobial stewardship and antimicrobial resistance. Therefore, the labeling or the removal of the penicillin allergy or a drug allergy is now a known weapon in the fight against superbugs and antimicrobial resistance. I was fortunate enough to be actively involved in a multidisciplinary team involved in removing these allergy labeling so that the labeling in hospitalized patients was able to witness the positive impacts on our cohort of patients. So let's now try to understand how a penicillin allergy label or a drug label has an impact on our patient's hospital stay. Here we have a patient that enters the hospital with a penicillin allergy, for example. During his hospital stay, this patient might unfortunately develop an infectious episode. Because he was prescribed broad-spectrum antibiotics, this person can have another unfortunate adverse drug reaction. When the patient leaves the hospital, he will have gained new adverse drug reactions. So what can we actually do when a patient reports a drug allergy? One of our clinical in vivo tools that we use on a daily basis on the allergy clinic is skin testing. Following negative skin tests, we understand that more than 85% of all penicillin allergy labels can be removed. Our gold standard in allergy, and drug allergy particularly, is drug ingestion challenge. And this test involves giving a test dose to our patients. If the patient develops no adverse drug reaction, we can then remove the allergy label or what we just mentioned, the delabeling process. In our cohort of low-risk drug allergy patients in the hospital setting, we have managed to demonstrate that 96 to 99% of low-risk penicillin allergies can be removed by oral challenge. So are there actually any clinical scores that could help us risk stratify drug allergies? The PenFast tool is an international and internally and externally validated clinical decision rule. This tool has been developed by our group led by my mentor, Dr. Trubiano, and the risk of a positive allergy test can be accurately predicted using this clinician decision rule. As you can see on the right side, with a score of less than 3, this is associated with 5% chance of developing an allergic reaction. And this actually brings us to our trial, the PALACE study, or the use of a penicillin allergy clinical decision rule to enable direct oral penicillin challenge. This is the first international multi-center non-inferiority randomized controlled trial. Our main question is, do we still need to perform skin testing for patients with low-risk penicillin allergies as defined by a PenFast score of less than 3? As I mentioned, this is an international trial. With our site in Canada, we are fortunate to collaborate with 2 centers in the United States. And finally, in Australia, where I participated in the design of the study during my fellowship, and I am very fortunate to currently be part of the leadership team. Our patients are currently being randomized to either skin testing, followed by oral challenge if skin testing is negative, or direct oral challenge. The randomization is currently stratified by clinical site, and the sample size that we aim is 380 patients. The recruitment is ongoing, and we have managed to recruit 351 patients today on both arms. So we consider that the ability to deliver point-of-care penicillin allergy investigations for this large cohort of patients without skin testing will improve patient access to testing and the use of preferred penicillin antibiotics. The data will be collected from all the centers that are implicated and will be analyzed together, and we hope that this will lead to local practice change, but also be considered as part of new drug allergy guidelines. When I am not focusing on labeling the 10% and fighting antimicrobial resistance, I am very passionate about severe confirmed allergic reaction. My focus is to actually focus on the true allergic reactions and develop ex vivo immunological assays in the lab. The aim of these tools is to have no risk of drug re-exposure for our patients, as well as evaluate drug probability and guide safe drug reintroduction. In our work, we managed to demonstrate that a combination of in vivo, such as skin testing, and ex vivo diagnostic tools in the lab can help in the context of severe delayed immune-mediated antibiotic adverse drug reactions, can help improve causality assessment. I had the opportunity to study various broad-spectrum antibiotics, such as piperacillin, tazobactam, Bactam, amoxicillin, clavulanic acid, and currently Septra, in order to understand how we can improve ex vivo tools in the immunological lab. So based on the data from the PALACE trial, how can we improve hospital care and optimize care for our patients? How can we optimize the use of critical care medicine by improving antimicrobial stewardship? In an ideal hospital setting that you can see on the slide, the patient with an allergy label would be assessed by an inpatient surveillance program as soon as the patient is admitted to the hospital. This will lead to an antimicrobial stewardship evaluation and hopefully the removal of his drug allergy label before the patient actually developed his infectious episode. If the patient did end up developing, unfortunately, an adverse drug reaction following the prescription of his medication, this would be followed by another antimicrobial stewardship evaluation in order to guide appropriate use of antibiotics and appropriate use of medication for the rest of his stay and for future hospital stays. Unfortunately, if the patient will leave the hospital with persisting drug allergies, this will lead to an outpatient allergy evaluation where we would have the possibility to optimize our in vivo skin testing and ex vivo immunological assays in order to help predict what medication led to that adverse reaction. Over the following years, I really plan to understand translation and implementation research in drug allergy and how we can optimize drug allergy diagnostic tools. So as we discussed, allergy delabeling is a critical component of broader antibiotic stewardship interventions. I do hope I managed to convince you that removing a penicillin allergy offers the potential for enormous impact on both patient outcomes and healthcare costs. Defining safe and accessible pathways for penicillin allergy risk assessment and testing is essential to address this significant public health issue. Dr. Cook is an inspiration to researchers worldwide, as her studies have transformed critical care medicine and have led to an improved global healthcare that is focused on patients, practice, and policy. As penicillin allergy is a significant public health issue in a high-burden population, I am also optimistic that our research will have long-lasting impact on the management of drug allergy. And I would like to take the time to thank all the wonderful people that are making drug allergy research a better place. I am surrounded by amazing mentors and collaborators, both locally and internationally. Thank you very much for your attention.
I can't tell you how needed that research is as an internist and practicing critical care physician. That is pain we experience every day, and it certainly impacts care. So I applaud her for her efforts. Our next speaker, I have the pleasure of introducing on a personal level. I am very thrilled to introduce Dr. Deborah Cook. She doesn't know that I'm here, that I'm wearing this hat today, but she's actually my personal mentor. She trained me in critical care medicine and certainly guided my career in so many immeasurable ways. She is such a humble and kind person. She actually visited me in Oakville several times over the last 10 or 15 years just to provide personal support. So I'm just shocked that I get to have the privilege of introducing her today. She is the 2022 Canada Gairdner Award for pioneering research that has developed and defined evidence-based critical care medicine in Canada and informing best practices around the world. Guilty admission, if I go to an ICU conference somewhere in the world, getting a lukewarm reception, I'll just drop, I know Dr. Cook, and the room changes. Trust me, I've used that one a few times. She's also the Distinguished University Professor of the Departments of Medicine and of Health Research Methods, Evidence and Impact at McMaster University, Fellow of the Canadian Academy of Health Sciences, Fellow of the Royal Society of Canada, Officer of the Order of Canada, and a critical care physician at St. Joseph's Healthcare in Hamilton. Thank you very much. It's my honor to introduce Dr. Cook.
It's great to see Sunny here, and Anna, thanks for a beautiful presentation. Let me start by thanking the Gairdner Foundation, the committees, peer review, individuals, the board, all the folks, the staff who've helped to underscore the important role of science in society. And I wanted to reflect for a moment on the exhilarating week it has been as a scientist in our country, meeting high school students, parliamentarians, early career investigators, mid-career scientists, senior scientists, just talking about our shared quest for knowledge. I'm humbled to stand among all the amazing scientists being honored here today and receive this award myself on behalf of the staff and the students with whom I've had the honor of working, my clinical colleagues at the bedside, like Sunny, many across the country, and the collaborators and co-investigators who've helped to enrich my career. These are some of the female staff, scholars, students with whom I work in Hamilton. I was so delighted to grow up in part at McMaster University, where questioning was part of the way we do things, and where equity and diversity and inclusivity were in existence before those were even the EDI three-letter acronym. Eminence-based medicine was alive and well when I graduated, and we had the great fortune of David Sackett, who came from the University of Chicago, who told us all about clinical epidemiology and the power that it held to transform the way we practice by offering insights and ensuring that we would learn whatever we could about the basic and clinical investigations that were published that would hopefully inform our practice, not dictate what we do, but enlighten us. Gordon Guyatt was my amazing mentor who struck that special balance between challenging me and nurturing me. A tightrope to walk, and one that I've tried to do with other individuals who I've had the honor of mentoring. At McMaster, there were some amazing scientists. Roberta Bondar, neurologist, Canada's first female astronaut, and someone who I know has also inspired others. Roberta being out there with her meteoric career, pardon the pun, was, even though I didn't know her, a source of inspiration. And May Cohen, a University of Toronto graduate, who was big in labeling women's health and calling to attention women in science. So this was my home. I was interested, though, in a vulnerable group of patients, not those in a clinic, not those on the general wards, but those who were critically ill, individuals requiring basic or advanced life support, individuals on the brink of death every day, individuals requiring much more than a doctor to look after them, nurses, respiratory therapists, physiotherapists, occupational therapists, pharmacists and nutritionists, so many individuals, social workers and spiritual care clinicians. This was not a community in science as well developed as in the States, and I went to Stanford University. Welcome to the modern world of critical care medicine. Overwhelmed by technology, you'll see that it's difficult to identify a patient in the middle of this image. But the patient has been the inspiration for all of my work, the patient and the family, and that holds true for critical care clinical investigators across our country. We began in the mountains. Coming back from California for a weekend, a group of us got together in Alberta. We were funded with a massive grant from MRC, $1,500. Go ahead, knock yourself out, get together. How about forming a research group? We didn't know what to do with that money, so we rented a bus and some beer, and we drove into the mountains and discussed together how it was we were going to collaborate. We took some inspiration from the view, and we realized as a large country with a small number of individuals, it was going to be important to collaborate. We needed more rigorous studies. We needed larger studies, 200 patients, 1,000 patients. That was not enough. We wanted to collaborate coast to coast, and we had to develop more infrastructure, individuals who were actually skilled in how to enroll patients in observational studies and randomized clinical trials. We knew it was important to ensure the quintessential Canadian camaraderie, and we thought hard about our mission. Bold, simple, and empowering, to try to advance the care and improve the outcomes of critically ill children and adults. This was through not just clinical research, but excellent clinical research. And it was important back then, and we didn't need a multi-million-dollar company to come in and do visioning with us. We said, what are our values? Well, we obviously are going to value scientific creativity and integrity and ensure that what we do is clinically relevant. And we shortened that pipeline from the early days of laboratory-based and animal-based investigations to try to enhance the possibility that studies, when they're done, they would be ready to consider implementing very soon after completion. We were very clear on the need for collegial relations. We had a little mantra, check your ego at the door. Some people were left behind the door. And some joined us after they learned the way we do things. We were always keen to ensure we were accountable to each other. We critiqued each other's protocols. We wanted to be responsible to our whole community and be fiscally responsible, knowing how precious resources could not be squandered in the early days of a research group. We were talking last night about organizational culture in the business world and how all of us have affinities for certain cultures. Organizational culture could be defined loosely as how things are around here, the invisible forces that move people to act, to behave, to speak. And we're all part of different organizational cultures in our communities, our place of work, our home perhaps, places of faith, in gyms. But the organizational culture that we wanted to be part of, and I personally was keen to create, was one that valued innovation and creativity, was always rational and strategic, was group-oriented, but was not rigid, hierarchical, sexist. And knowing that this organizational culture could take us far, I think those of us involved in the early days wanted to ensure we retained some of these values. So our first chair was Dr. Thomas Todd, a thoracic surgeon, who got us together in the feisty days when people were a little edgy about getting together. And I was so honored to be a two-term chair of the trials group, followed by a wonderful friend and colleague, Dr. John Marshall here in Toronto, a trauma surgeon intensivist. Paul Hebert spent most of his time at the University of Ottawa, and Robert Fowler here in Toronto, who's been an incredible gift to critical care through the pandemic, and Dr. Kusa Menon in Toronto, a pediatric intensivist. So our organizational culture really helped to define how we do things. So I'm going to speak a moment about foundations of clinical programs. So because in a large community, when we had nowhere to go but up in terms of generating our knowledge, it wasn't one project. We weren't going to do one-off little projects. We had a lot to sort out in the field. We were able to create ideas, do a little bit of early design, but it was through the connections and the conversations that our projects really took roots. The design features were key. We debated, we discussed, we poked, we prodded, and this notion of discussion and honest reflection and sharing our views was fundamental and has still been fundamental to this day about our clinical research programs. I'm just going to give you one example, and this is about gastrointestinal bleeding from the proximal gut, basically the stomach. And it's a kind, stress bleeding, as it was called back then, was related to bleeding from the stomach, related to allegedly the thin lining of the gastric mucosa and impaired blood flow. And we had noted that in the early days, people who were critically ill tended to be having a lot of bleeding. It was quite frequent, it was often fatal. And in fact, our patients were sometimes receiving 2 different drugs to try to prevent this bleeding. And we thought that was a little bit odd. And this example of a research program shows how critical it is to not just jump in and do a trial, because we were all ready to go. We're going to do a randomized trial. Let's try to see, are antacids better than this other drug? So I thought that maybe we were ready to do that right away, but wisely and upon reflection and discussion, we went ahead to do some additional preliminary pre-trial observational studies. So a prognosis study to try to identify what the risks of bleeding would be, what the incidence was, and actually come up with a definition. Turns out to be quite important. It's not live or die, it's what is bleeding that is clinically important. We went on to examine, does bleeding really matter? What's the attributable morbidity, mortality of bleeding? Is it something we should be concerned about in terms of prevention? And then, of course, before the trial, just re-estimating what the relative risk reduction might be if we were to do a trial. And we went on and did this large trial, but this preparatory work was a crucial lesson for our community about the need for observational studies in humans before we launch a trial. And this work was the foundation of our early success in New England Journal of Medicine, and the randomized trial likewise was in New England Journal of Medicine. And this was something that was very important to us to understand all the problems that can be averted if you carefully conduct observational studies to get all your ducks in a row before you launch an expensive RCT. And these are just some of the many lessons that we've taken forward for the Canadian Critical Care Trials Group. Interestingly, this randomized trial did show that the use of an antacid, at this time was ranitidine, was associated with a 50% lower bleeding rate than a weak base, this agent called sucralfate, which was widely used and is hardly ever used now. But there was also a suggestion of an increased risk of pneumonia in patients receiving this antacid, the so-called gastropulmonary route of infection. And after the trial, we further used the RCT database in an observational analysis kind of way to determine risk factors for pneumonia and understand the attributable morbidity and mortality for pneumonia. And this launched a whole other career of Darren Hyland, who took this forward in a diagnostic RCT, also published in New England Journal. So some of the messages from these integrated research programs that stayed with us included the idea of not one-off studies, which can be certainly super important, but as we've heard about this morning, the idea of the long view. And we had this agenda that could be rolled out and would give incremental bits of knowledge that would take us forward. And Katie also underscored that earlier. So we had lots of questions. We needed to have methodologic advances because a lot of the critical care literature, modest as it was in patients, was not all that rigorous. and our funding was scarce. So we began to understand we need to harmonize. Everyone can have different ideas, that's great, but let's make sure we synergize around questions. We don't necessarily prioritize what we do, but we try to energize from getting together and make sure we mobilize as a community to be clear we can conduct what we set out to conduct. Second brief example I wanted to review is about blood clots. I'm sure everyone's familiar with blood clots in the legs or arms, sometimes the pelvis, which can break off and cause blood clots in the lung. And for those patients who are critically ill on mechanical ventilation, they have poor cardiopulmonary reserve. And a blood clot going to the lung in that instance can be potentially serious, not to mention hard to diagnose. So knowledge generation is key to critical care research in Canada, but so is faithful, timely knowledge translation. And that can involve applying the results of our research, but can also, and sometimes must, be associated with deprescribing and omitting practices that we thought were suitable. So the triad for blood clots is stasis, associated with immobilization, and obviously critically ill patients are super sick in bed, often heavily sedated, sometimes paralyzed, especially in the era of COVID. Vessel wall injury, because inserted catheters, peripheral, central, dialysis catheters, ECMO catheters, this is very, very common. And critical illness, this is a pro-inflammatory, procoagulant state. So it's like a perfect storm for blood clots. So like all good scientists, when we were beginning, we thought, all right, let's do an environmental scan. What's written about blood clots for critically ill patients? So we looked to some of the practice guidelines populating the literature. I was shocked that medical surgical ICU patients were not even mentioned anywhere. So, as you know, when there are holes in the literature, this is one of the good signals work needs to be done. And we embarked on another big program, but the first that I wanted to point out was the way in which Canadians reached out very early to do international collaborations. And this was a simple comparison between colleagues in France and Canada. There was the standard unfractionated heparin, and then the newer, more potent, and slightly more expensive low molecular weight heparin. And you can see the self-reported utilization confirmed when we observed what happened in an audit, completely disparate practices, which is, again, another good sign of, oh my gosh, this needs to be studied properly. So we went on with colleagues, not in France at this time, but Australians, investigators in the US, the UK, and Brazil to do the PROTECT study, and in fact, tested the low molecular weight versus unfractionated heparin comparison. And once again, the idea was, let's go, let's do this trial. No. We thought, we have more work to do. And we were so grateful to work with our translational biologists because there was some discussion amongst our colleagues about how there could be pro-thrombotic markers like factor V Leiden, prothrombin mutations. Some of these measures in the blood could be excellent predictors of blood clots, and perhaps we should be doing some genetic testing and try to identify a high risk and potentially responsive population to enroll. And that turned out not to be as fruitful, but was a crucial part of the journey. So very much later, after PROTECT was done, we thought, all right, what are the results and how are we going to translate them into practice? So the primary outcome was deep vein thrombosis of the leg, diagnosed by ultrasonography twice weekly. And you can see there that the hazard ratio, if it's 1, would indicate no difference one way or the other. And there was only a trend toward fewer blood clots in the legs. Pulmonary embolism, or PE, looked like a 50% reduction in PE. And no overall strong advantage in terms of reducing mortality, but HIT, the heparin allergy, seemed to be reduced about 50%. And importantly, no increased risk of bleeding in this higher risk, newer, allegedly more potent heparin. So what do we do with a study like that? Well, people in France were saying, yeah, so you should be using it. And other folks are saying, yeah, but it's more expensive. Is it really all that much better? And we did go on with a number of other studies, but with the help of Rob Fowler and colleagues at Sunnybrook, we thought this again is a perfect situation where you're balancing all these things. And to recap, although the rates of DVT were similar, maybe slightly lower with low molecular weight heparin, certainly less PE and less HIT, the pulmonary embolism, the HIT, were very high morbidity, high mortality, and costly events, though rare. And it turned out that despite the cost differential in drug acquisition, when you cost out beyond the drug acquisition and think about the cost of these events, these clinical events in a trial, it was pretty clear that the low molecular weight heparin was a better way to go. And even then, they're just words. And we thought, what's going to speak to people in terms of knowledge translation? We've heard earlier about it's not just the individual practitioner, but their health policy implications. Other people are involved in which drug to use. And the analysis that we did identified that over a year, if there was a center that wanted to keep on using the unfractionated heparin, and they admitted 1,000 patients, they'd waste $1 million. Well, that caught everybody's attention. And so a lot of our work now in knowledge translation is trying to understand how to express what this might mean. And a lot of our work with patients and families made it pretty clear that everything that could be bad associated with these two different types of heparin was a similar probability or a lot less likely or a little bit less likely with the low molecular weight heparin. And so some of the family input was super helpful here. I want to end with one other example. As you know, Canadians want to largely die in the comfort of their home if they have a home, with family members, if they have willing family members, if they can afford it, and if the situation is right. Most people die, most of us in this room will die in an institution of some kind, potentially, likely, technologically supported. Although life support can save lives, really, it just delays death. Death is still very common in the ICU, and I know all of you are well aware of that, having been through the COVID-19 pandemic. A good death, according to the World Health Organization, is a human right. That is not a reality for many places in the world. With our universal healthcare system in Canada, we hope that it could be a reality. We were interested in the early days in studying how people die in the ICU. And way before the pandemic, high proportions of patients were dying. Whether they came in with trauma or a surgical condition or medical condition, people were not really wanting to talk about it. And I recognized early on this was a place where there are numerous knowledge gaps. And I remember a few senior authorities in my world thought that this was really not a suitable area to study for a senior or junior scientists with any sort of credentials or brains. And I was actively discouraged from studying this. And in fact, one of my colleagues really wondered if it was ethical to study this. And I just felt sick hearing that. But I thought, it's unethical not to study this. This is how Canadians are passing away. And it was always a favorite focus of my practice and aspect of teaching. But we kind of put this theme on the slow burn, started involving nurses who are delivering the minute-to-minute daily care of critically ill patients, and involved several folks who were keen to take this forward, did a survey, and then did an observational study. And this was something that we wrote a grant for $400,000 from the Ontario government, and they awarded it, and only they awarded $40,000. So I remember writing to them saying, did you maybe forget the zero? Because it's 40th this morning. He said, no, and it was a clear message. This is not, this is not so important. But we went ahead. A lot of people volunteered their time and energy toward this. And starting with 851 mechanically ventilated patients, as you know, when the ventilator is weaned or people are liberated from the ventilator, they breathe on their own and hopefully live. But you can see that there's still a proportion who die in ICU, die in hospital, and go. There's some go home. Some people die on the ventilator. They just don't get better. And then there are people who we withdraw from mechanical ventilation in anticipation of their death. And that might not be imminent, but it happens eventually, for the most part. But as you can see on the bottom there, we don't always get that prediction right. Some people anticipated to die actually leave hospital. And this was quite illuminating, sobering, you might say, and led us to wonder what the predictors are of this withdrawal of mechanical ventilation. I know everyone in our community and folks participated in this in Australia and in the US, thought it was going to be pre-ICU hospital features like old age, really sick, a lot of comorbidities. None of those were predictors. The predictors were the additional need for advanced life support, these medications which keep the heart rate up and the blood pressure up, and patients who are predicted to have a very low survival, which is largely a doctor's notion. And then people who were predicted by physicians largely to have impaired cognitive function. And then elicited from the family preferences. The family told us that these patients would be probably not wanting to live like this if they were going to survive. And this last notion helped to underscore the importance of the family input when we were discussing goals of care. And this was also the first time where we asked people to share their views about what their concerns were when people were on life support, often for days and days and seriously ill, because obviously many get better, but many do not. And this was the lens of ethics on end-of-life care, the uncomfortable lens of discussions of morality that were initially not welcome in the day. So what did we learn here? Engagement of the bedside staff was key, and we did surveys along the way to ask people's views. This really promoted some self-reflective practice, which was key, and because it was largely self-funded, it signaled to funders how important this was, and was a way to legitimize end-of-life research, if you will, and led to many conventional metrics of research success. So for this theme, if you will, the idea here of the passion of the topic, but your patience and waiting till the time is right to really launch a program was key, and persevering. And this led us to recently do a number of studies on something called the 3 Wishes Project. The overall objective was to try to improve the dying process so that in the fast-paced, technologically intense, dynamic, sterile setting of the ICU, it was possible to honor the inherent dignity of dying patients, humanize the dying process for family members and support them in grief, all those who are left behind, and then to try to inspire a deeper sense of vocation for the clinicians caring from them. And the idea was these terminal wishes could be anyone's wishes or preferences, the clinicians, the families, the patients, if they're able. And our hope was that this would bring comfort in the final days. Many wish categories exist: bringing in pets from home, playing music in the room, a taste of someone's favorite wine under the tongue, keepsakes, which have been very important in the pandemic, a number of different wishes. What is a wish, really, at the end of life? It is a process, it is a product, and a way to have a conversation starter for the individuals we care for. We often forget who are they before their critical illness and beyond their critical illness. These are some of the ways we conceptualize what is a wish in the 3 Wishes Project. We found through many multi-center studies with colleagues here in Toronto, Vancouver, UCLA, Brantford, Niagara, and a number of other cities in North America, as this has been taken up in a lot of different places, this approach to honoring the dying patient helps to recognize the inherent dignity in all human beings, gives voice to the family to try to create some memories, and calls forth clinician compassion, still a very important part of critical care. Fast forward to the pandemic, family members prohibited from being in the ICU. Covered up, so everyone looks like a Martian. Dying at these times, this was tough. We wondered, can the Three Wishes Project help us? And we were ready. We found that clinicians were inspired to learn each patient's story, to try to advocate, step in where family or friends could not. The use of video conferencing technology, which has been used obviously everywhere, under appropriate circumstances can help to connect when people cannot be together and surrounded by the things and the people that they love. So amidst the challenges of providing personalized end-of-life care during the pandemic, clinicians were able to express their humanity and support patients and their families. So although in critical care we have more and more technology, we still need to figure out how to use it, in whom, when, and how long. Humanism has been at risk, attenuated sometimes, but we've always been keen to ensure that recognizing inherent dignity is maintained. And I'm now hopeful after the pandemic and all of our observations that humanism is alive and well, as is dignity-conserving care in critical care medicine in Canada and hopefully worldwide. Thank you so much.
I think it should be obvious to you why she is a mentor for so many of us in critical care and why she's literally a global leader in critical care medicine. So thank you again, Dr. Cook. It is now my pleasure to invite early career investigator Dr. Bo Wang to the stage. Dr. Wang is assistant professor at the University of Toronto, CFAR AI, or Artificial Intelligence Chair at the Vector Institute, and lead AI scientist at the University Health Network's Peter Munk Cardiac Center. Dr. Wang was selected by Drs. John Dick and Dr. Stuart Orkin for his work on machine learning-based approaches to development trajectories of hematopoietic stem cells. So thank you. Please, Dr. Wang.
Hello, everyone. I'm very honored and deeply grateful for this amazing opportunity and award. A special thanks to Dr. John Dick and Dr. Stuart Orkin for their nomination and recognitions. I often introduce myself as a computer scientist who happens to be interested in biology. Today, I want to talk about how machine learning or artificial intelligence can help advance our understanding of hematopoietic stem cells using single-cell multiomic data. So, first of all, quick question. What is the first thing appearing in your mind when people mention artificial intelligence? I guess probably it is like this for some people. However, in reality, AI is much less cooler. Instead of threatening people's life like alien, AI is actually helping improve people's daily life. For instance, it has been widely used in smartphones to understand our voice, or it is used in autonomous driving cars to free tired drivers. AI, like no other, has struck our life like a storm. Without little doubt, we are entering a new era where AI is greatly advancing the pace of science as well. Notable examples include AlphaFold, an AI tool by Google's DeepMind to predict 3D structures of protein folding, a biology problem unsolved for more than 50 years. AI is also being used to expedite the drug discovery process, and relevant to my research, AI or deep learning is shaping many single-cell data analysis or many other genomic data. But one may wonder, what makes AI so successful? My solution to you is very simple. I summarized as ABCD as an analogy to sports cars. So if we think of AI as a sports car, to make the car run really fast, you need 4 things: great wheels, strong engine, articulate steering, and sufficient oil. Same thing applies to AI. We need these 4 components as well. Here, A, algorithms are the engine. B, business is the steering wheel. C, computing is the 4 wheels. And last, D, data is the new oil, or electricity if you want to be friendly to the environment. So in a more academic setting, AI is a very wide concept in which machine learning is really a subset of AI. Further, deep learning, the very algorithm behind all the hype of AI, is just a subset of machine learning. So what is deep learning? Deep learning is an algorithm that bases on multi-layer neural network, which I show on the right. In it, you have a bunch of artificial neurons that can be set on and off, and there are connections with neurons. The cool thing about deep learning is that it can automatically adjust these millions of weights by showing lots of real-world examples. The question I ask here is that how can AI advance the understanding of hematopoietic stem cells, or HSC for short? The project I want to talk about today is really motivated by a few recent works by both Dr. John Dick and Dr. Stuart Orkin. Specifically, Dr. John Dick's group recently proposed a cellular hierarchy framework that combines both bulk RNA-seq and single-cell RNA-seq to understand the heterogeneity of AML patients. Further, to fully grasp the differentiation process of HSCs, both groups suggest that we should go beyond just transcriptomic signatures by including chromatin structures or some other epigenetic data. Therefore, we propose a new approach which bases on novel data-driven method, specifically machine learning, to integrate single-cell multi-omic data to further understand the heterogeneity of ML and to infer the developmental trajectory of HSCs. As I mentioned before, a recent paper by Dr. John Dick's group in Nature Medicine proposed a new ML hierarchies which deconvolve single-cell RNA-seq with large cohort of bulk RNA-seq data from ML patients. And the defined cellular hierarchies can well explain many clinical heterogeneities such as drug sensitivity, genetic mutations, and survival progressions. Moreover, with this tool, we can now project single-cell RNA-seq data of any AML patient into a normal hematopoietic development map and understand the patient-specific trajectories, moving single-cell genomic one step closer to the clinical adoption. However, the newly proposed cell hierarchy still does not fully solve the heterogeneity of AML patients. For example, if we project a collection of public single-cell RNA-seq data from AML patients into these 4 hierarchies, we still see quite large heterogeneity among these 4 clusters. So this leads to our aim 1. Can we do a better job in dissecting the ML heterogeneity by integrating other genomic data, such as RNA-seq, such as DNA methylations? What we have done so far is take RNA-seq data and cellular compositions of about 800 ML patients and DNA methylation of a subset of about 300 ML patients and develop a novel machine learning algorithm called SAM2 to provide a comprehensive view of ML heterogeneity. SAM2 is a data-driven machine learning algorithm that is based on 2 of my previous approaches published in my lab. The advantage of SAM2 is its ability to merge a collection of non-overlap genomic data in an unsupervised manner. If you recall, we have RNA-seq data for about 800 ML patients, but a methylation only for about 300 patients. So traditional integration method will have to take the intersection of the 2 modality by discarding the non-overlapped RNA-seq data. However, this is not an issue for our SAM2. After SAM2 integration, our method finds 12 distinct subtypes of ML. We can see that these 12 subtypes have very different cellular hierarchies and transcriptomic patterns, but also different genomic pattern. Further, we verify that among the 300-ish AM patients, the methylation profiles and the metabolic pathways are quite distinct too. Clinically speaking, these ML clusters have significant different survival patterns and drug sensitivity. We also map these 12 clusters onto single-cell map of normal hematopoietic lineage and find distinct marker gene enrichment pattern. We then further validate these 12 clusters onto some other public ML cohorts, such as TCGA and BEAT ML. And again, we find significantly different survival progressions. All of these validations give us confidence about our machine learning approach and the validity of the found 12 subtypes. The second aim of this project, which is still an ongoing work, is to integrate single-cell multi-omic to have a comprehensive view of continuous developmental trajectory of HSCs. Specifically, we will develop a deep learning algorithm called DeepVelo to combine both single-cell RNA-seq and single-cell ATAC-seq from multiple HSC conditions in order to learn the RNA velocity in a data-driven approach. Once the model is trained, we will open the black box and use sensitivity analysis to interpret the trained model and perform multiple analysis to identify relevant transcription factors that may drive the self-differentiation of HSCs. So the deep learning system named DeepVelo represents one of the first deep learning-based attempts to learn cell-specific kinetics, and is currently under review. DeepVelo had already been successfully applied to learn the RNA velocity of human cerebellum development using single-cell data. The success in brain data gives us confidence that this approach will also be helpful to HSC multi-omic data. Last but not least, I want to conclude my talk by summarizing that machine learning, or AI, really represents a paradigm shift in modern genomic research. Traditional approach usually is hypothesis-driven, while machine learning-enabled approach many times is data-driven. Traditional approach often work on small data set from single lab, but machine learning-enabled research approach requires large-scale data with multi-center validation. Computationally, traditional bioinformatics often use stepwise pipelines that are specific to each task, while machine learning approach tends to work on general-purpose framework with carefully designed end-to-end framework on more advanced hardware such as GPUs. With the prevalence of AI, collaboration between computational scientists and biological scientists are ultimately important than ever before. Last, I want to thank again Dr. John Dick and Dr. Stuart Orkin for their foundational work that inspired my research. Also, many thanks to my amazing mentors and collaborators, and certainly all the credits go to my trainees and students. Thanks for listening.
Thank you, Dr. Wang. Next, we will hear from his mentor, Dr. Stuart Orkin. Dr. Orkin was awarded the 2022 Canada Gairdner International Award for the discovery of the molecular mechanisms responsible for the switch from fetal to adult hemoglobin gene expression during human development and translating that knowledge into a novel treatment for hemoglobin disorders, sickle cell disease, and beta thalassemia. Dr. Orkin is a David G. Nathan Distinguished Professor of Pediatrics at Harvard Medical School and Dana-Farber Boston Children's Cancer and Blood Disorders Center, and an investigator at the Howard Hughes Medical Institute at Boston Children's. Thanks. Please welcome Dr. Orkin to the stage.
Thank you very much. I appreciate the recognition for the Gairdner Foundation, and want to also thank the Foundation for the wonderful week we've had, and congratulate the other laureates for this year. So one of the challenges we like to think about as we apply molecular biology is how to use these tools of modern genetics, not only to understand biology, but also to formulate mechanisms of mechanism-based therapies that are aimed at really transformative clinical outcomes. And that's what I'm going to be discussing in the work today. I'll be reviewing. I've had an evolving career in which I've had the good fortune of living through many transformations in biology, such as recombinant DNA cloning came on the scene as I began. Also, prenatal diagnosis of disease by DNA, positional cloning of disease genes, stem cell biology or regenerative biology, genome-wide association studies, and most recently, genome engineering. And you'll see facets of this in the work I'll describe today. So I've been interested in blood cells for my entire career, where these cells come from, how do they form, how do they go awry in disease. And the context in which we've done much of the work is that of red blood cells. How are they programmed? What goes wrong in disease? And can we do anything about what we learn in terms of therapy? The two major disorders of hemoglobin are sickle cell anemia and beta thalassemia. This is sickle cell anemia, first coined as the first molecular disease by Linus Pauling in 1949, and then 7 years later, Vernon Ingram identified a single amino acid change at position 6 of the beta chain of hemoglobin. That's the cause of sickle cell disease. And this is a sort of a classical view of beta thalassemia, which is a disorder not of the protein coding region itself, but that of production of beta globin, where one gets this classical phenotype in untreated individuals. I should emphasize that these are global disorders with a health burden of increasing magnitude. I think the incidence map that you see on the left resembles a map you've seen previously, actually, of childhood deaths. And it's, there are at least 300,000 new births a year of individuals with sickle cell disease, and there are about 75 to 100,000 individuals currently in the United States. This is really the crux of the biology that we've focused on over many years now. On the top is chromosome 11, which depicts the beta globin locus, which is more than 60 kilobases in length. We've had the DNA sequence of that for more than 35 years, and none of that DNA sequence actually helped really understand the mechanism of regulation. What happens during ontogeny is in embryonic life, we express an embryonic gene epsilon. During fetal life, the fetal genes, which are the gamma genes that are duplicated, and in adult life, the beta globin gene, which forms beta globin, expresses beta globin, which is one of the components of the tetramer of hemoglobin. The mutations of sickle cell anemia and beta thalassemia all are within the adult beta globin gene. And I should mention that as adults, we have about 1% fetal hemoglobin at steady state. And as you can see, around the time of birth, there's a progressive switch from gamma to beta, which is the so-called hemoglobin switch. Now, in the mid-'80s, early 80s, we worked with Hay Kazazian, who recently, unfortunately, passed away, working out the mutations that cause beta thalassemia. And at the time, we thought we'd learned something unique about how beta globin is expressed, how red cells form. In fact, we didn't really learn that. What we found that there were many different mutations, but most of these affected generic functions of a gene, such as RNA splicing, nonsense codons, transcription, and they didn't really provide the kind of insights that we were seeking at the time. This information, however, was very important in terms of establishing prenatal diagnosis of hemoglobin disorders in many centers around the world. And this was probably the first disorder for which we had a complete molecular understanding. In order to get back to hemoglobin F, though, we have to look at some of the early sort of insights. And one of them came from a pediatrician, Janet Watson, in 1948, who noticed that infants who were born with sickle cell disease, at least the genotype, were not sick in the newborn period, but became ill 6 months or so after birth. And she surmised that this might be due to the elevated fetal hemoglobin they still had before the switch was complete. And in fact, she was correct. Fetal hemoglobin is protective both in thalassemia and in sickle cell disease. And a natural history study done through the NIH many years later showed that individuals with higher fetal hemoglobin on average with sickle cell disease live longer. Besides the fact that increased fetal hemoglobin replaces a beta globin because the expression is reciprocal of these genes on the chromosome, hemoglobin F has the unique property of also disrupting the polymers that form of the sickle hemoglobin. So you get sort of 2 bangs for the buck. So the field as a whole has wanted for really essentially my entire career to want to increase that 1% to some therapeutic level of fetal hemoglobin, which was estimated to be about 20% per cell. That's a rough number, but it's somewhere in the ballpark. We then tried to understand how are globin genes transcribed. And this led us to a transcription factor called GATA1, which was actually the first hematopoietic transcription factor. And it's the master regulator for red cell gene expression. Every gene in red cells depends on this factor. It's a relatively small protein, and it binds a specific DNA sequence. The problem with GATA1 is it's not stage specific. It's expressed in embryonic, fetal, and adult life, and therefore it cannot explain how this switch occurs. And really the conundrum for quite some time has been how does the locus control region, which is a powerful enhancer for the locus, the whole locus, how does it engage the gamma genes in fetal life, and how is that switched to the beta gene in adult life? What proteins control that switch? Well, we've had a long history of study of fetal hemoglobin going back, for example, to Janet Watson in 1948. Many different studies of globin genes and globin gene expression as soon as one could begin to clone genes. But around, I'd say around 2000 or before that, we were in a desert. We didn't understand what factors regulated fetal hemoglobin, and we were coming up dry. And many of us were actually working on other problems at the time. And then we needed something new. And I love this quote from Marcel Proust, which says, "The only real voyage of discovery consists not in seeking new landscapes, but having new eyes or new technology." And the new technology that appeared was genome-wide association studies, which are studies of common genetic variation and how they correlate with traits or disease across the genome. And there were 2 studies done, one in the UK and one in Sardinia, and we were fortunate to collaborate with those investigators in Sardinia, basically looking at fetal hemoglobin measurements, the level of fetal hemoglobin in populations, the very low level 1%, sometimes it's a little higher, and these studies resulted in this so-called Manhattan plot, which is what geneticists who do GWAS call these associations. And since I was born in Manhattan, I put the Empire State Building over the most important gene, which is the new gene at the time, which was BCL11A on chromosome 2. These studies also identified chromosome 6, the MIB gene, which is an oncogene controlling cell differentiation, and the beta globin complex itself, which was not a surprise. What is a surprise is summing these 3 genetic contributions accounts for at least 50% of the genetic variation, the level of hemoglobin F, which in these kinds of studies is a very large number. The problem in genome-wide association studies is going from 3 billion bases in the genome to understanding something about what you aim to study in the first place. The first question I want to address, though, is how important is B11A? I mean, we didn't know whether there were 10 factors, 100 factors, or one factor controlling the split switch. And this experiment is probably the most illustrative, in which we used sickle cell mice. These are mice in which the mouse genes are ablated by homologous recombination, and human genes have been inserted and expressed relatively normally, and they have a sickle mutation. And we interbred these with a mouse that had a conditional knockout for BCL11A, and we restricted the knockout to the erythroid lineage to avoid any confounding issues of other cell types. And we published this actually in 2011. And the results of this experiment were much more striking than we would have guessed, predicted at the time. This is a sickle mouse. It has sickle cells. And here's the interbred mouse. It has an entirely normal blood smear, normal hematology, has lots of fetal hemoglobin, and it's distributed in all cells. And this level of fetal hemoglobin is above this 20% level I suggested before. And these mice were perfectly normal and live a normal lifespan. So that told us that one gene, if we manipulated it, could be curative, at least in a mouse. The BCL11A protein is a big protein, about 100 kilodaltons. The business end is over here, which are zinc fingers. They bind DNA, and they bind a unique sequence, TGA-CCA, which had never been identified for a transcription factor previously. But remarkably, it turns out it's in the field promoter, and is also the site of mutations, which are very rare mutations leading to fetal hemoglobin in adult life, suggesting these mutations disrupt the binding of the protein. The proteins are a repressor, therefore the genes are expressed. And these were reported more than 35 years ago, and Francis Collins was actually a postdoc at the time who reported one of these. And these were totally unexplained for more than 30 years. So how does this protein work? I don't have time to go into all the details, but basically in the field stage, there's an activator called NFY that binds the gamma promoter and drives expression. And then in adult life, BCL11A is expressed. It's not expressed before the adult stage. It bumps off NFY, evicts it from the promoter, we think structurally, and drives repression. And what's remarkable about this is this action, this switch occurs over a platform of about 30 nucleotides, which is really the major guts of the whole switch process. So the promoter itself has evolved to be the sort of template for the switch. So what about this GWAS? I told you how the protein works, but I didn't tell you how the genetic variation factors into this. Well, it turns out that the BCL11A gene is a large gene, but the SNPs, the genetic variation that correlated with hemoglobin F levels, is actually within a large intron right in the middle of the gene, where there are 3 DNase hypersensitive sites in erythroid cells. These generally connote regulatory elements. These are not present in brain and B cells where the gene is also expressed. So that suggested this was a red cell specific element. And in fact, in transgenic mice, it behaves precisely that way. And we suggested in 2013 that we could use this element really as an element for genetic modification because it's absolutely cell specific. And if we modified it, it would have no effects on gene expression of this gene in other tissues. We then went on, once CRISPR actually came on the scene, this was in 2015, so we were early adopters of the CRISPR methodology. We actually peppered the enhancer region with a series of guides to ask where were the functional components of this enhancer. The enhancer itself is 10 kilobases in length, but we identified actually an Achilles heel. I call it dumb luck, but it sits at a GATA site. The transcription factor we identified in 1989 is important, And cutting this one site, a single cut, leads to about a 2/3 to 3/4 decrement in expression of B11A. And so that immediately made it obvious that that was the place one needed to use gene editing. So we had gone from the whole genome of 3 billion base pairs down to really one GATA site, which we could exploit therapeutically. So how could we do that? Well, there were several ways to think about manipulating B11A. One would be to knock out the gene. That's not a good idea because it's toxic to hematopoietic stem cells and you lose B cells. You could knock it down, and that's actually a trial ongoing at Boston Children's Hospital led by David Williams, a colleague. But the one that's gone the furthest is to edit the B11A gene at the enhancer sequences at Achilles heel. You could also mutate the binding site in the promoter, and that's ongoing as well in clinical trials. What B11A is, it's basically a rheostat or thermostat for hemoglobin F. And the question is, how much do you have to dial it down? Well, nature gives us somewhat of an answer. This is an unfortunate circumstance for individuals who have haploinsufficiency. That is, they have one chromosome in which B11A is either deleted or mutated. They have a cognitive deficit, auditory speech deficit, somewhat of an autism-like syndrome, but they have 50% B11A. And the question is, what's their hemoglobin F? Well, their hemoglobin F is about 10 to 15 times normal. So that's essentially approaching the therapeutic range. So the bottom line of this is you don't need to dial the expression down tremendously. You can dial it down 50% or a little more, and that's in the therapeutic range. So how do you approach this genetically now? And this is the current state of gene therapy of blood disorders, in which one takes hematopoietic stem and progenitor cells, modifies them ex vivo, so it's done in the laboratory. It's then these modified cells are introduced back to individuals, the patient whose bone marrow has been conditioned, it's been destroyed by chemotherapy, and then the new cells reconstitute the hematopoietic system. There are many facets, parameters are important in this. I don't have time to describe them, but I just want to emphasize that every one of them is important in carrying out a successful gene therapy trial. So what is the current state of what's being done? It's to take advantage of the Achilles heel in this 10 kilobase enhancer and make a single cut at that GATA site, disrupt 2/3 or so of the activity of this enhancer, and this single cut impairs B11A expression, relieves repression of fetal globin genes, permitting increased fetal hemoglobin. So how well does this work? This is not our work. This is the work by a collaboration of CRISPR Therapeutics and Vertex Pharmaceuticals that have actually taken one of the guides from a publication and put it into action in both thalassemia, beta thalassemia, and sickle cell disease. And this was a publication in actually December of 2020, describing the first patients. And you can see in a patient with thalassemia, essentially 100% F. These are because they can't make beta globin. And in the sickle cell patient, they make about 40% F. And the bottom line is patients are well. They've extended this trial. This is the latest public information with 31 patients with sickle cell disease, all of whom are now free of crises, and they're free of any need for transfusion. And these patients are probably cured as best we know. And this trial has been submitted, initial submission for FDA approval. So it'll probably be the first CRISPR therapy approved for a genetic disease. So we believe the genetic approach has been validated, but this, as Drew said earlier, this is an expensive procedure. It's going to be $1-2 million for each procedure and cannot be exported widely to many patients. So what can we do about that? One is to improve preconditioning, make it non-ablative, perhaps with antibodies. That's undergoing study by a number of laboratories. The other approach is in vivo gene therapy, which Drew Weissman hinted at, possibly combining lipid nanoparticles with gene editing. And I think here the question is, it's only going to come down to the efficiency and the scale up, whether it's actually doable. So just to summarize, over the past more than 15 years now, we've gone from really the universe of genome-wide association through to understanding the mechanism of how this switch occurs, which was sort of an enigma for my entire career, and then using either gene editing techniques or lentiviral techniques, it's possible now to downregulate B7A for a therapeutic result. And we think this will have positive impact, obviously, for many individuals. I just want to sort of emphasize also the timeline of this, that we started with fetal hemoglobin maybe in 1948. We had a long gap where we didn't know how to bridge that gap, and genetics has sort of gotten us across that now into what we think is anticipated approval of a therapy. I just want to emphasize in closing that this is a global disorder with a real health burden, and really we have to focus in the future on trying to reduce that health burden in order to really make an impact in disease. I have a few take-home messages. One, we understand this switch pretty well, but I think an important point is that really B7A has now been thoroughly validated as a therapeutic target genetically, which I think will hopefully stimulate further interest in vivo gene therapy, using this as a target. And now that we have a mechanism for the switch, we can think about rational drug design, perhaps as an alternative approach. Many individuals have contributed to this. I don't have time to do justice, but I just want to show many who have contributed to this portion of our work. And then finally, I want to give thanks to my family, my wife Roz, who's here, and daughter and grandkids. Thanks very much.
I want to thank Dr. Orkin. I can tell you as a practicing physician, that work was seminal in the reality we live in today, where there's 4 approved gene therapies by the FDA for treating previously untreatable conditions. So it's absolutely outstanding. I now want to introduce our final early career investigator lecture of the day, which will be given by Dr. Professor Yannick Benoit. Professor Benoit is Assistant Professor in the Department of Cellular and Molecular Medicine and School of Pharmaceutical Sciences in the Faculty of Medicine at the University of Ottawa. Professor Benoit was selected by Drs. John Dick and Dr. Stuart Orkin for his research on the epigenetic mechanisms affecting cancer stem cells in colorectal tumors. So please, let's welcome Dr. Benoit.
Hello, everyone. First of all, I would like to thank the Gairdner Foundation for giving me this opportunity to be here today and to present how 2 of this year's laureates of the Gairdner Awards positively influenced my career path as a scientist. So I want to start with this picture of me when I was at the very beginning of my journey as a PhD student at the University of Sherbrooke. At that time, I was working on a research project involving some key transcription factors, including GATA transcription factors, by the way, and their role in the differentiation process of human intestinal epithelial cells. And through my readings, I bumped into the great work of Dr. Stuart Orkin during his stem cell biology phase, I would say, based on what he just told us earlier. So Dr. Orkin's work was oriented on specific chromatin regulation mechanisms, including the polycomb repressive complex 2 and its role in maintaining a balance between self-renewal and differentiation in embryonic stem cells as well as in blood cells. And this concept, like, I applied it to my current work, and this led to interesting papers which inspired me to pursue a first postdoctoral fellowship at the Weill Cornell Medical College in New York City, where I kept on working on these epigenetic and chromatin regulation mechanisms, but this time in the progression of colon cancer. And this is where I got more and more interested by a concept that was developed by through the outstanding career of Dr. John Dick, the concept of cancer stemness and hierarchical cellular organization within the tumor. So not all the tumor cells are equal. In fact, there's a hierarchy that is governed by a small population of cancer stem cells. And these have critical functions such as self-renewal and tumor initiation capacity. So at that time, I was starting to think about my own independent research program, and I wanted to put a translational component into it. So I wanted to go and learn more about drug discovery. And then I came back to Canada and joined the lab of Mick Bhatia at McMaster University, where I started to study stem cell-based assays that could be incorporated into drug screening assays. And then I wanted to develop as an independent program a drug discovery endeavor, I would say, where I would find new therapeutic tools to target cancer stem cell populations within colorectal tumors, but by aiming specific chromatin regulatory pathways. And this is what I'm doing since 2017 at the University of Ottawa. So in my lab, we are interested in several chromatin regulation pathways and their role also like in cancer stem cells. One of these pathways is the role of the histone methyltransferase G9A, which is overexpressed in a certain subset of colon tumors. And we've shown last year that G9A is important for pluripotent-like identity and tumor-initiating functions in colorectal cancer. So just to mention specifically that G9A is catalyzing the deposition of two methyl groups on lysine 9 of histone H3. And this epigenetic mark is typically associated with transcriptional silencing. So we and others, we've shown that G9A is important in colon cancer to maintain self-renewal functions, but also tumor initiation capacities. And other groups recently demonstrated that G9A is key to promote immune evasions in tumor cells versus the immune system. So while G9A may sound like an exciting new therapeutic target, well, we must keep in mind that G9A is also very important to maintain homeostasis in numbers of normal tissues. So that's probably why, so far, among all the small molecule inhibitors that were developed to specifically block the histone methyltransferase activity of G9A, none of them actually made it to the clinical development stage. So in the lab, we're trying to develop safe pharmacological approaches to target G9A function in cancer stem cells population, but we want to do this with limited harm as much as possible in normal tissues. So one of the strategies that we're using is drug repurposing. We are starting from clinically approved compounds or compounds that are in late stage of development, for which the specific target and the mechanism of action has already been documented, most likely. But most importantly, like those compounds already have demonstrated clinical safety. So they were proved as being safe when they are used in human subjects. So in one of our projects, we applied a phenotypic drug screening pipeline to which we subjected a library of clinically approved compounds. And this pipeline is a succession of two stem cell-based assays. So the first one was developed by Saklos and collaborator in 2012, and is to measure the capacity of our compounds from our library to decrease the expression of a pluripotency marker that is called OCT4. So whatever drugs we have that is going to decrease OCT4 expression in transformed embryonic stem cells, we're going to move these candidates to the next stage where we're going to measure the capacity of those drugs to decrease the deposition of the G9A catalyzed mark H3K9 dimethyl. And out of the candidates that we got a few interesting ones. There is this mystery structure here that we're going to call for today, YB1471. So YB1471 is not only decreasing OCT4 expression in transformed embryonic stem cells, but it's also simultaneously decreasing H3K9 dimethyl. And when we treat three different colon cancer cell lines with this compound, we saw a dose-dependent decrease in cell viability. And we were not observing this effect when we tried the compound on human normal intestinal cells. So it suggests like a selective toxicity impact that we have toward the cancer cell lines. So to make a very long story short, we've shown that the drug is decreasing, in fact, the protein levels of G9A, and this is due to an impact on the promoter of G9A per se. So YB1471 is in fact an antagonist of the dopamine transporter SLC6A3, which is going to bind to this transporter via the same binding pocket than cocaine. And when we look at the distribution of this transporter in colon adenocarcinoma cross-section, we saw only a handful of cells that were presenting positive staining for dopamine transporter across the section. And this result was confirmed by single-cell RNA sequencing, where also we could only detect the transcript of SLC6A3 in very few cells of the tumor fraction, and nothing was observed in normal epithelial cells. So when we knocked down this transporter, we saw that colorectal cancer cells were having a decreased clonogenic activity. And this is reflected by a decrease in the organoid formation frequency in knockdown cells versus the control vector. So at the level of G9A protein levels, we saw that just like our drug, the knockdown of SLC6A3 was also leading to lower level of G9A expression. So we wanted to test the impact of our drug in the cancer stem cell compartment at the functional level. So we started with an assay ex vivo. This is a patient-derived serial tumor organoid formation assay that we reported earlier this year in a publication, where we start with some primary tissue, so primary tumor samples that we grow in non-adherent conditions to enrich cancer stem cell populations. And then we dissociate these resulting spheres, I would say, and then we seed at the single cell level in a 3D setting these patient samples, which will form a primary series of organoids. So we will treat these organoids with our drug of interest versus a control. And then after 14 days in culture, we will take the resulting organoids, we dissociate them again, and we replate them in a secondary series of colon tumor organoids. And without any further drug treatment, we're going to check whether or not we have a change in the frequency of tumor organoid formation. And I bring your attention to the secondary organoid data that we have here on the slide, where we observed that our drug treatment was causing a drastic decrease in the number of secondary organoids formed compared to the vehicle control. And interestingly, we also used a drug that is commonly used in the clinic to treat colon cancer, called 5-FU, and we could not observe such an impact on tumor initiation capacities by treating our primary organoids with 5-FU. And to confirm that G9A was really involved in the mechanism of our candidate compound, we ectopically reintroduced G9A expression in colon cancer cells, and we saw a rescue of the effect of our drug at the level of tumor initiation functions. So because the gold standard assay to measure cancer stem cell activity is to go in vivo, in vivo serial tumor transplantation model, we've done exactly the same exercise than what we've done in organoid, but this time in mice recipients. And so we engrafted colon cancer cells in a primary series of mice, which we treated during 10 consecutive days with our candidate compound. And then we dissociated those resulting tumors and re-engrafted secondary recipient series. And what we saw at the level of the secondary recipients is, again, we have a strong decrease in the frequency of secondary tumors that are formed in the group treated with our drug of interest versus the vehicle control. So To summarize, I've shown you that using a stemness-based drug screening approach, we were able to identify a clinically safe inhibitor of G90 functions in colon cancer stem cells in vivo. And I forgot to mention that the compound was not causing any harmful effects when we look into the normal tissue in the gut. Also, our multiparametric screening strategy, as I presented it earlier, did not only highlight a new therapeutic tool, which would be our drug, but it's also pointed toward an unexpected potential new biomarker and new druggable target in colon cancer stem cells. And finally, if we consider the translational potential of our compound of interest, because it's already been deemed safe in human subjects, our work opens on new tangible possibilities to alter critical epigenetic pathways, such as G9A, in a therapeutic context. So finally, I would like to give an extra special thanks to all the students in my lab, as well as my past scientific mentor, and the key collaborators to this project. And thank you very much for your attention.
Thank you, Dr. Benoit. Our final laureate lecture of the day will be given by Dr. John Dick, recipient of a 2022 Canada Gairdner International Award for the discovery and characterization of leukemic stem cells, providing insights into the understanding, diagnosis, and treatment of acute myeloid leukemia. Dr. Dick is a senior scientist and Canada Research Chair in Stem Cell Biology at the University Health Network's Princess Margaret Centre and University Professor in the Department of Molecular Genetics at the University of Toronto. Please welcome.
Well, I am truly honored to be included in the 2020 class of laureates, especially including the illustrious scientists and people you've heard from this morning, and in particular, Stu Orkin, who is just a towering giant in the field of molecular hematology, and so to be included on the same stage is truly an honor. So what I want to do today is to give you a story. It's a little bit hard to think about, going through 35 years of work in 30 minutes. And as some of you know, I've talked, I'm not one to include less slides than I need to. And so we'll see how well we do here. We might have to skip a few at the end, but that's all right. I am the last speaker, although I do recognize you've been sitting for 5 hours, and I'm standing between you and lunch. So what I want to do is to really address the topic at the very top, which is the simple one, which is what makes a stem cell a stem cell. It was an old question that came in the 1960s. And we still have it as a question today. And of course, how does it go bad? And I want to cover these themes that you're going to see in front of you here. So, it's appropriate to give you the context of the field as I started when I was working in it. And that is, I did my postdoc with Alan Bernstein and mentored by Bob Phillips in the mid-1980s, I guess. And of course, 25 years before that, Tillen McCullough had brought forth the modern era of stem cell research with this defining experiment, caused, in fact, by some serendipity, but amazing insight. What they realized was that when you transplant cells into a mouse and you look 14 days later, there are these nodules on the spleen made of about 1 million cells. And what they realized was that each nodule was the product of a single cell. And so what they recognized then was that a single cell can go through, and they looked at the property of the cells, and they were multi-lineage. And so all of the multi-lineage you see in the nodule there must have come from a single cell. And that redefined the textbook, because in the 40s, people knew there were stem cells, but they thought there were stem cells for each lineage. And what they were doing here, and why I said it was the birth of modern era of stem cell research, is they were counting stem cells. They were quantifying them. That was the big advance. And from there on, we started to understand stem cells in better ways in all systems. Now, the assay is based on something which is a paradox, and it still exists today. And that is, stem cells are these powerful cells that are in your blood, in your bone marrow. They are used 50,000 times a year at least around the world in a stem cell transplant for cancer therapy. Powerful cells, but we don't know how to study them if we put them in a lab dish. And so you can only study them by watching what they do, and what they do is to regrow the blood system. And of course, that's all well and fine, and so for 25 years before we started, people have done a lot of work to define these in mouse systems and then infer this must be how the human works. Now, the problem is I wanted to study the human system after I left Alan's lab. Oops, wrong button here. And so the question was, how are we going to study them? Well, others had gone on and made colony-forming assays. So some cells here have progenitor-like activity, and they make colonies in a lab dish. But the question is that truly the stem cell? In the mouse, we knew that wasn't. These can only be assayed by transplantation. Now, nobody wants to volunteer for my experiments. So the question is, how are we going to study human stem cells? Fortunately, I was working with, my great mentor is Bob Phillips, who's sitting right over there, who was a pioneer of working with immune-deficient mice, naturally arising mouse that doesn't have a T cell and a B cell, doesn't have an immune system. And so we sort of thought, well, maybe if we just put, we put human cells in a mouse, but if it doesn't have an immune system, it's not going to reject them. And it turns out there's enough complementarity between what a mouse can provide and what a human cell needs. You do this experiment, you have mice running around the cage that have some of their portion being, some of their blood coming from human cells. And that set off, a 25-year pathway to understand what is the needle in the haystack. I like the cartoon that Stuart had. What is the needle? What is the one cell in a million that has the ability to do this powerful job? And so that took, a lot of work. And I need to say that this work was done by Suzanne Campbell-Mead, who was my very first postdoc, that, somebody coming into the lab would take on this crazy project of putting a human cell in a mouse, and really optimized by Steve Lapidoux, who I'm pleased is sitting in the audience here, who really brought this on and made it work for the world. And this has become the gold standard now around the world on how to study stem cells. So we and others contributed to how this tree in 2005, particularly the Weissman lab at Stanford, had brought in this marker 90 positive and negative, and they thought that was the true stem cell as opposed to this cell down here. And I had two amazing students in the lab at the time, Sergei Dulatov and Fayez Nada, who looked at the published data from the Weissman lab, and they realized that there were stem cells in the 90 negative population here. They're much less than the top, but they're actually this. And so the question was, there must be some marker that we seem to be missing. And what they looked at was integrins, because in the mouse system, we knew integrins could capture different kinds of blood stem cells. My colleague Norman Iskov was also able to see this in the mouse system, and so we were guided by that. And so we looked across the human system and realized that one of these integrins, integrin alpha-6, CD49F, is actually expressed differentially in both of these populations. So it's not just expressed in the, the enriched stem cell population, Phi positive, they was also present here. And so that set off the search. We wanted to see, could we find stem cells in this pot? And we went to the ultimate experiments, a lot underlying this one experiment, but essentially we sorted all these cells, added in something which just captured dormant cells, and then we literally sorted cells, looked visually coming off the sorter in a well, pulled them up in a syringe, and injected that single cell into a mouse. And what we had then was a mouse running around that had blood in it, and it had blood that was on the myeloid lineage and the lymphoid lineage. So that was the defining, two major arms that you can define a blood stem cell as giving. And if it gives both, that must be a multipotent cell. And we did this as single cells. We just counted up the mice that were engrafted and the ones that weren't. And it turns out that somewhere between 1 in 3 to 1 in 8 mice in the two different experiments that we did were actually engrafted, which meaning that we had achieved the best purity that had existed to the day, and actually still exists than today. We still actually need more markers to get that last 1 or 2 cells. So we get this to be 1 in 1. But we went a long ways to understanding what is a stem cell. As postdocs went on and studied this in more detail, we wanted to really understand the different populations of cells, and so we used timing as a way to characterize these cells. So we transplant, all these cells are multipotent, they'll all give multilineage, but some of them will do it more transiently, others do it more permanently, and then the definitive test is if you take these cells and put them into a secondary mouse, the canonical hallmark of stem cells, which is self-renewal, and we realized then that there is this hierarchy that we can then capture. And so over the years, and in fact, just in the last 3 years, COVID has actually been very good to us because we actually stopped and wrote a whole bunch of papers. But essentially what we've learned then by just studying the top cells at the top of the hierarchy, that long term and the short term, the key difference about what makes a stem cell has to lie in distinct epigenetic regulation. So with the advance of technology and single-cell ATAC and high-C and so forth, we've understood that there is a unique 3D chromatin architecture around 280 sites that are open in a dormant long-term stem cell that have to become closed down in order for that cell to exit dormancy and to become activated and enter the blood system. We've learned that these cells are uniquely designed for stress. Most cells in the body, if they get stressed by DNA damage or proteostasis, protein folding, misfolding, they will pause and try to repair it. If it goes on too long, then they'll finally kill each other or kill themselves. What stem cells do is if they're damaged in any way, they have very high levels for killing, and so you don't want to passage damaged DNA onwards, and so actually they cull. And that becomes a core feature of what makes a stem cell a stem cell. And then there's some organelle biology, some beautiful work showing that lysosomes are incredibly active in a dormant cell. A dormant cell isn't just a quiet cell not doing anything. A dormant cell is actually an incredibly programmed state, and one of the programs is around organelle biology. High levels of lysosomes that are always clipping receptors off the surface of this dormant cell, so you can't actually get a signal. So it's got a physiological way of generating dormancy. And then other aspects of metabolism. The one that I would just point to is one of these studies, and this is all done by careful single-cell analysis, amazing work from these postdocs, and this is the work of Kirsten Kaufman. So we thought that this long-term stem cell, 1 in 3, 1 in 5, is as homogeneous as we can get. And what you realized is that there is actually some heterogeneity in this stem cell. They're still the long-term stem cell. It's just that when you transplant them, one gets going very quickly and generates the blood system when you transplant it, and then it exhausts. So the activity that long-term stem cell has to generate quickly comes at a cost. It can't self-renew the stem cell. Whereas there's another population in here that we can mark based on this particular marker, which when you transplant, it takes a time to get going. But in that process, it actually preserves itself, and it then is the one that sustains itself long-term. Now, this is what happens in a mouse, but it turns out that we can know that this happens in humans because of the gene therapy trials that Stuart was mentioning. Some of those gene therapy trials, we can follow insertion sites of many thousands of stem cells, and these kinetics are exactly what happened in humans. So we think that we've identified a new class of stem cell and a new feature then of of stemness, which is this term latency. As a hallmark of stemness. And there's a molecular mechanism that underlies that's in this paper, and I can't talk about it right now. And so, like everyone, all of what I've showed you has come into single-cell biology. Now we can do, single-cell gene expression, single-cell ATAC-seq, and so forth. And the problem, though, in the blood system is that everybody can generate nice, fancy maps. You do single-cell sequencing, and you can cluster, and you say, oh, this population is erythroid, that's stem, and so forth. And, here's another one, and here's another one. The problem is all of these came from bulk analysis. And I told you, stem cells are super rare. They're probably, the long-term stem cell in these, there's probably no more than 1 or 2 long-term stem cells in any of these. So you can have a nice clustering and say, here's stem, but the question is, what is that? It's contaminated by a lot of other cells. And so it becomes a challenge then to try to interpret this data. And so Andy Zhang in the lab went forward. We basically amalgamated all of the data that we had, and then we could use our own data to help annotate where the stem cells are. And so by aggregating all of this, it has a much more balanced map. And we now have the best atlas on human cell development, including all of them, lineages down, the B cell lineage, the erythroid, and the myeloid down here. But of course, our interest is in the stem cells here. And so how good is that? We have this label here, HSC. Well, how good is it? Well, so we've done some work out of our own lab. So the data from Kirsten had sorted a bunch of single cells. And you can see all of the single cells of that phenotype all land in that little quadrant. This is another work, I'll show you in a second, but this is 5,000 cord blood sorted LTHSCs. They all land exactly in this quadrant. And then here's work from Lisa Levente and Dominique Bonnet's labs. By the way, both former postdocs in my lab. We all, have drunk from the same Kool-Aid and still do the same kind of experiments, but they all land there. So we can have a lot of, and these are all functionally defined. In all these cases, not only have these cells been analyzed this way, but we've been putting them into mice, and we know that all the repopulating activity is present there. And so, one of the things that Andy went on to do is he looked at this, what we thought was a homogeneous population, And by looking at the various pathways that are active by chromatin and gene expression, we realized that there was heterogeneity within what we thought was a pure population. And what really struck us, and this has actually come through 3 of the studies, this came from our epigenetics study, this came from our metabolism study, came from our lysosome study, that there is a subset of stem cells that seem to have high levels of ER stress inflammation. Now, these are cells that are lying dormant, right, in their niche. Why do they have these pathways going? And when you look, these pathways are driven by the classical NF-kappa B and ER stress pathways that are all localized down in this quadrant. And so the question is, what are these guys doing there? And I'm going to show, so what I wanted to do, by the way, I missed to say, what I want to do is to give you a perspective of where we came from, you know, why I'm standing on this stage, and then where we are today. And so this is now the new unpublished stuff. And so driven by Andy and Taz, who are student postdoc in the lab, but really guided by Stephanie, who's an affiliate scientist in the group, who this is her baby as we're moving forward with a lot of collaborators. And so what we've come to realize is, guided by the work in the mouse system, is that inflammation is a huge biological consequence, a consequence of biology. Everybody is going to experience this inflammation. We often have viewed inflammation, and as I'll show you, inflammation memory, on the basis of the mature immune cells, right? Memory T cells, memory B cells. We all understand that. We are vaccinated as children. We don't see an antigen for 40 years. We see it, and then the memory cell gets going, and we make an antibody. But it turns out that work from in the bloods on the murine side, and carried out actually largely by Emmanuel Passaguet, who is in the audience, had shown that actually the sensing can occur at the stem cell level. So when stem cells see an inflammatory signal, they get activated because it's like an emergency response that they need to go. And but of course, you have inflammation, you don't lose all your stem cells, so you must have to have a recovery. And the question is, how does this happen? And in particular, how does that happen in the human system? And so, these things are elusive. And the interesting thing is, this isn't just an inflammation thing, but if you look at the consequences of an acute inflammation compared to a chronic, the chronic really impairs the stem cells, and it really creates this myeloid output. And that's a feature of aging as well. So there's an aspect, we talk about inflammaging, right? So inflammatory aging processes are happening at the level of the stem cell. If that goes on too long, we can see signatures of this stress in the early steps of malignancy. And so the question is, how is this all happening in the human system? Largely what I've told you here has come out of the mouse system. And so Stephanie and the group set up this experiment. We took human cord blood cells, very naive population, whacked it into immune deficient mice, waited 6 weeks, hit them with a single dose of LPS or TNF, and you can see we got the classic drop in engraftment 16 hours later, the classic acute response. And the question then is, well, what about the recovery? So if we wait for another couple of weeks and look at the mice, we can see that they recover just beautifully. And we know in our xenograft model that all these dormant stem cells get very activated by 2 to 4 weeks. So most of the stem cells are actually cycling at that point. And in this particular case, we did a second challenge, but we can actually do this with a single challenge. But I'll just show you this data. And then what we do is we wait for 20 weeks. So 2.5 months later, we say, did that have any consequence, right? They recovered. But in fact, if you look here, there's actually a permanent consequence to these cells, even though they only saw the exposure, single exposure, a couple of months earlier in these cells. And so the question is, where is it coming from and how is this happening? So we undertook multi-omi analysis. This is a single cell ATAC, combined ATAC RNA sequencing. And if we wouldn't have done this, we wouldn't have been able to capture what I'm going to show you next, because RNA sequencing is not very good at the top end of the blood system because there aren't enough differences. These cells are too similar. But it turns out that, ATAC, as my good colleague Matthew Lupien always tells me, epigenetics is a much better predictor of cell state than gene expression, because it predicts what's going to happen in gene expression. And so by having the core embedding and know what our stem cell signatures look like, we can actually interrogate this data in a much more robust way. And what that told us was there actually are there's one population of stem cells which we reported on, we've come to love and know, and it's at the top end of the whole blood system. We can see trajectories here using Bo Wang's various trajectory prediction tools. But it turned out when we look carefully, there's actually another population, I'll call it HSC2 here, marked in green. And what was interesting is it seems to go up with the response to TNF. So what I'm just to tell you is, I'm not showing you the cord blood itself, but just putting these cells into mice expands this. We can see rare evidence of this in cord blood, but if you put it into mice, you see an expansion of this particular stem population. And so the question then was, well, how are they changing as a consequence of the exposure to TNF? So we compared that population to that one. And what became clear is that there is a whole set of gene expression differences which are higher in the LSC2 compared to the LSC1. There's a set of peaks that are much higher in the LSC2 compared to the LSC1. And of course, you can integrate that data and predict what the transcription factors are. And all the top transcription factors up here are AP1, June, the FOS June and the AP1 transcription factors, as well as other classic immune regulators. They're all in the HSC2. They're not present in the HSC1. Very distinct stem populations that pre-exist here. This is just a summary of a lot of data, but essentially what this shows is we seem to have one homeostatic population that just sustains hematopoiesis, and it's not very responsive to the TNF inflammation at all. And then there's another population which is very present and responsive to this inflammatory setting. And so the question then becomes, what happens? Is this relevant in the human setting? So we made a signature of that, we call it HSC inflammatory memory. We made a signature and we asked, how does it compare across this aging spectrum? So this is bone marrow taken from people at different ages, and you can see the signature really increases with aging. So this is part of the human, this HSC memory population must be a part of human aging. And this is just another independent data set from two different groups of young versus old bone marrow. This is pediatric and adult. Again, it fits with the idea that this is not just a feature of a crazy experimental model, but in fact, it's a feature of human aging. And it seems then that HSCs themselves are incredible biosensors of this inflammatory activation. And the question is, what about leukemia? And there's a beautiful paper that was published by Irene Papia-Petru from Sinai in New York, where they had created an engineered model of human leukemogenesis. And they would introduce single genes to create the clonal hematopoiesis, the pre-leukemia step, and then the more aggressive, and finally the AML. And at each stage, they were able to model and ask what are the kinds of signatures that underlie the difference between a normal stem cell and this primed pre-leukemic stem cell. And the key features that come up here are all inflammatory regulatory transcription factors. And these are the factors that come up exactly in our model here. So we think that there's actually a link between this new stem cell that we've uncovered and the clonal hematopoiesis that underlies pre-leukemia and pre-leukemic expansion. So I think there's a lot of, we're quite interested in obviously understanding this in much more detail. So that's the normal side. So cancer. If I ask this audience or I stand out on the street corner and ask people walking by what's cancer, I'm always going to get the same answer, which is cancer is cells that grow too much. Cancer is cells that grow in an uncontrolled way. That's undoubtedly true. But if you frame the question in a slightly different way, and that is every cancer cell equally able to keep that cancer going? If a cancer cell is born today and it dies the day after tomorrow, it's really of no relevance, right? And we know that tumors are dynamic entities. Cells are always growing and always dying. And so the question is, what's the cell that keeps going, both keeps the tumor going, but also obviously in the face of therapy? And that's the question that interested us from the beginning. After we had put normal human cells into mice, the question is, what about leukemia? And the paradox I told you earlier is exactly the same one, and that is, these are leukemic cells in the bloodstream of a person growing rampantly, and they're going to kill that person, but I still can't take them in a lab dish to study them because I still, even today, don't really understand the signals that they need, and they need an in vivo environment. And since we'd already put normal cells into mice, we were able to put leukemia cells into mice. And it turns out that, that works beautifully. Mice will run around with human leukemia and they'll end up killing them. And then the question was, how many cells can do that? And we carried out this limiting dilution experiment, and it turned out that only 1 in a million cells did that. So this is really weird, right? 1 million cells, if they're all equal, why is it only 1 in a million? Well, it could be that this is just a crappy assay, or it could be that there's a rare cell there. And people had already identified a colony-forming assay, McCullough and Metcalf and others, about 20 years before we did these experiments, had already shown only 1 in 100 cells is clonogenic. All the other cells are post-mitotic progeny. And so is this just an inefficient colony assay, or is there an ancestral cell that's earlier than a colony-forming cell? And this is the work that, Zvi, if those of you who were at the symposium yesterday, gave a beautiful summary on. But this is, the reason that I'm standing on this stage is because of the work that Zvi Lapido did in the lab, both to optimize the normal assay, but really to carry out this experiment. And what we had to do then was to ask, if it's just a crappy clonogenic assay, we knew that there were clonogenic cells in this quadrant up here and some over here. And so the question is, can we separate our activity of putting leukemia in a mouse away from the clonogenic cells? And so we sorted these samples into a pot down here and a pot up here. This actually represents, most of the cells. This is the 99% of the cells that represent the bulk of the tumor. This is only 1% of the cells down here. We whacked them into mice, and only these guys gave leukemia. This is the data from the original experiment. Each dot is a mouse. This is human cell engraftment. These cells up here never gave leukemia, no matter how many cells we transplanted, where these guys gave leukemia in the mice. And so this proved that the cell that engrafted in a mouse is different than a clonogenic cell, indicating that there is a hierarchy. And that has gone on. I just point out we've had a wonderful trip here with Zvi and his wife in Petra some years ago. And so what that led to then was the idea that not every cell in a cancer is equal. There are some cells that have stem cell potential, and those are the stem cells of the cancer. And that turns out to be true not just in leukemia, but in other solid tumors, at least some solid tumors. Catherine O'Brien in the lab and Antonio Creso moved forward and were able to show that colon cancer is organized in a hierarchy like this. And then actually other Canadian scientists made a lot of contributions. And my good colleague Peter Dirks was a neurosurgeon at Sick Kids when I was still there. And he used our leukemia paradigm and brilliantly showed that brain cancer is organized too, which was actually one of the first with Mike Clark in breast. Those were the two papers 10 years after we did our leukemia work to show that actually solid tumors can do this as well. Okay, so what I've really told you then is that leukemia isn't just abnormal cells, it's actually a caricature of normal development. It is normal development. There's stem cells, there's progenitor, there's mature cells. And so if that's true, not just an artifact of putting human cells in a mouse, it must mean that these are the only cells that keep the tumor going. And if they're the only cells that keep the tumor going, they must be the most important in the tumor. And if they're the most important, they should be the most relevant. And so we had to prove to ourselves what's relevance. And so we thought, well, what clinical parameters could we use to evaluate whether leukemia stem cell properties are relevant? And so we thought, well, the only binary outcome that anybody will believe is whether it can predict whether you're dead or alive, right? So unambiguous outcome. And so what we did is we had to figure out what's the property of a leukemia stem cell. And we used gene expression, first by Kolya and Eric, and then later by Stan, Amanda, and Jean. But essentially, we took large numbers of AML patients, all independent patients that came from our biobank. Our brilliant collaborator, Mark Minden, was able to contribute for 35 years. We sorted them into fractions, did gene expression. We whacked them into mice because every patient is actually a bit different. They're not always leukemia stem cells here. They can sometimes distribute them differently. So put it into many, hundreds and hundreds of mice. Captured all those fractions from these 84 patients that had leukemia stem cell activity and those that didn't. Integrated their gene expression and came up with 104 gene signature that was differential between leukemia stem cell positive fractions and those that were negative. These were quite predictive of clinical outcome. We refined that with machine learning into a 17 gene signature, right? 17 gene signature of whether a human cell can engraft a mouse or not. And how does it do? Well, it does amazingly well. This is clinical data. We've actually worked with the molecular diagnostics lab. This is now a certified test. And if you're high signature, you do much more poorly than if you're a low signature. Remember, 17 genes out of whether a human cell can engraft a mouse or not. How artificial. Yet it means then that these stemness properties that signature is defining must be the coalescence of this is 300 ways to make a cancer cell, right? Everybody has their own private cancer genome. 300 ways to make a cancer cell, but this stemness property predicts how you're going to do, suggesting there's a lot of convergence going forwards. And so we're mining that. To continue on then, just to finish this part, what Stan wanted to do was think about, well, if this is, a caricature of normal development, again, an old concept from Barry Pierce in the 80s, can we use the resemblance of how any individual leukemia looks in relation to normal development? In other words, how much of a caricature is it? Is it a good caricature, a highly relevant, or is it just a little glimpse of it? And so you can think of it this way. There are leukemia stem cells, and sometimes some of those leukemia stem cells will just make a lot of cells that are just like another leukemia stem cell. Or otherwise, some of these leukemia blasts might have a lot of features of differentiation on them. We actually have known that from the 19th century. The hematopathologist looking at a microscope said, oh, there are leukemia cells that have more granulation than are others. And so they reflect, if you like, that development. So how can you capture this? Well, single-cell technology enables that. So Peter Van Galen reported in a beautiful paper, actually when he was at the Broad, but he was a PhD student with me, that he could take individual leukemia samples and do single-cell genomics and then map what they look like. So there are some samples which looked, very primitive-like in relation to their normal development. There's other ones that are more, mature, other ones that are more GMP-like, other ones that are more intermediate. I should say this is Andy's taking Peter's signatures and then mapping them onto our normal hematopoietic map, showing how powerful that map actually is. Bo actually presented this already, so this is just actually taking that data. There isn't very much single-cell data, but you could actually use computer deconvolution to take these signatures and mine bulk data sets, and that now accesses thousands of samples, and we could refine patients into primitive, mature, or GMP. And the question is, what does that do? Well, it turns out that if you're primitive, you have much poorer outcome than if you're GMP, where you have much better outcome, and mature are sort of intermediate. So there are clinical features that are relevant from knowing this hierarchy-based classification system. But the more important one is a hierarchy like this sensitive to the same drugs as a hierarchy like that or a hierarchy like that? And how do you do that? Well, it turns out that there is nice clinical data from a beautiful trial where they took many patient samples and they did drug screening for about 100 drugs on each and every sample as they came through. At the same time, they did sequencing. So we deconvoluted the data and then related the deconvolution score or hierarchy classification against drug sensitivity. And so this is just an example. Here are samples where if you have this kind of a hierarchy, this mature kind of a hierarchy, you're very sensitive to all these drugs, mostly signaling molecules and so forth, but you're very insensitive to the latest, greatest drugs like venetoclax and other molecules. On the other hand, if you're primitive-like hierarchy, you're insensitive to these drugs, but you're very sensitive to these drugs. So it's really the first biomarker just based on hierarchy classification, which is able to have this level of ability to not just predict prognosis and be sort of a biomarker in that context, but it's actually able to predict clinical outcome. Okay, if I just take, I'll summarize this because I know I'm just hitting my 30 minutes here. I told you I was going to talk a little too long. We also were very interested in this simple question, and that is, person walks into the clinic here, and how does there are going to be 4 or 5 mutations. This is AML, not a huge number of mutations. That day that they walk into the clinic, the question is, what happened to that patient the day before, the month before, the decade before? Where did that cancer come from? And what's the cell of origin? Does it matter? There's 4 genes here. What was the first one? Why did the first one get this system going and move forward? And then, of course, you can also learn what happens between diagnosis and relapse. This patient gets treated, the disease comes back. Where does it come back from? Where is the origin of relapse? And so Lorenz Slush brought genetics to my lab, brilliant postdoc who's now at the Weizmann. And we've contributed to lots of this work, but I'll just summarize it in this one slide. I know that people who work in solid tumors are going to kill me when I say this, but essentially, we've been able to characterize what I'm going to show you here by just taking a single needle or a single blood contribution the day the person walks into the clinic, and by studying carefully with advanced genetic techniques, cell sorting, we can figure out the ancestral cell that started the tumor is still there the day the person walked into the clinic, and that cell started 10 years earlier. It's remarkable. And so what we have, of course, is everybody starts with a normal blood system. We know that where the initiating mutations are, they started in a stem cell. This defines an entity called pre-leukemia. It turns out by looking at large population cohorts, we can actually track these mutations in normal populations more than 10 years in advance. This work, of course, contributed to the work that Ben Ebert talked about clonal hematopoiesis. We just studied the end of that, where people are going to go on and get leukemia. In that 10 years, they pick up additional mutations. They finally get a leukemia. Leukemia stem cells evolve here. But the key is, when we studied the diagnostic sample and compare it to the relapse sample, it turns out that the relapse fated cells are there the day the person walks into the clinic. So in the AML, all the bad things that happen have already evolved before the person walks into the clinic. And what chemotherapy does, it selects these relapse origin cells going forwards. But it means that we really need to understand this window, but we have 10 years to do it now, finally. And so the opportunity for prevention in a disease like AML is really quite remarkable. And in the last slide, I think 2 slides I have, the question is, we want to model this, right? It's fine to study human disease, but you actually want to know mechanistically how you do this. And it's all, it's difficult. You can't do this with primary human cells, or you can't do it with just a human leukemia sample. You need to start with a normal cell, put in the various oncogenes, and study the disease. Now, we were interested in pediatric leukemia, the beautiful work pediatric leukemia in utero. So you have a normal cell. A second hit happens and you finally get leukemia. And so the question is, how are we going to model that? And so this beautiful work that we followed the disease we followed was Down syndrome leukemia. So we know what some of the hits are. Of course, Down syndrome, they have trisomy 21. A high proportion of these children, 30%, actually have pre-leukemia when they're born. It often goes away on its own. Sometimes it needs a little bit of therapy, but it's a pre-leukemia, goes away. Then they pick up additional mutations, and about 20% of the patients go on and get AMKL, which is a very aggressive disease. And so what we know are some of the hits. GATA1, GATA mutation that Stu worked on. There's a mutation that causes a change in the isoform into a GATA1 short that's well known to be present. And then the other ones are cohesin mutations, STAG. And the question is, how do they relate to each other? What can we learn? And so this is work that Eric led, and Elvin Wagenblast, who's now new faculty at Sinai in New York, and Joanna Ariu was an MD-PhD student, did. And so we managed to capture, with our biobank colleagues at Mount Sinai, normal human fetal liver from disomic individuals, normal individuals, and also trisomy 21. We had quite a few fetal livers. We were able to use Elvin's optimized ability to CRISPR edit single long-term HSCs, transplant them into mice, and then carry out functional studies and ask what the consequences. So just in my last slide. So what do we know? Well, we know that if we engineer GATA1S into cells of different kinds, the only time we ever get pre-leukemia is we engineer it into a long-term stem cell, not even a short-term stem cell. It has to be a long-term stem cell. And it doesn't work if you put it into normal disomic fetal liver. It has to be a trisomy 21 cell. So we know it has to be a stem cell and it has to be trisomy 21. And Eric had been working on microRNAs that are LSC specific. There's 3 microRNAs on 21 that we can modulate, and they play a role in this process. So we actually know which part of 21 is actually playing a role in this, which has actually been a mystery in the field. And then the second one is, if you engineer the second hit in, it turns out that you can do any progenitor, or many progenitors down here, which means that the second hits happen in a lot of different progenitors, not just in stem cells. And then you get frank leukemia, and that's not restricted by, if you engineer both hits, you can put it into normal stem cells, but only in the fetus. If we do the same experiment in adult, it won't work. So there's something about the fetal environment, and that helps explain why pediatric leukemias are quite different than adult leukemias. It has to do with the cell of origin, but we now have a window to be able to get at it. And then I wouldn't be standing here if it wouldn't be for these individuals. I acknowledge some of them, but actually there's about 140 or 50 students and postdocs, plus a whole bunch of co-op students that have been there. And this is what I really want to say. This was a collective effort. We all have ideas that we throw into the crucible. None of us knows what our data means, but we talk about it. We throw ideas. There are good ideas. There are bad ideas. There's ugly ideas. Mine are often the ugly ones. But what comes out of that is the phoenix of the finding that I've shown you here. And I dedicate it to these individuals. And finally, I would not be doing any of this standing on this stage without my family. Jonathan and Becky and Chris and Dell and the 4 grandkids, Cameron, Grayson, Beckett, and Karis. I would not be standing here, and I dedicate this to them. Thank you.
Wow, what an incredible day. Thank you to our laureates for sharing your award-winning research. Thank you to our ECI winners. Clearly, the future of scientific research is bright. And my thanks to our chairs, Leah and Sunny, and to our Gairdner Science Week sponsors. Gairdner's always looking for feedback, so please fill out the survey at the end of the digital program and let us know what you think. Gairdner Science Week is coming to an end, but please follow us on social media at Gairdner Awards to continue the conversation. So for the students who have pre-registered for the student lunch, you can grab your lunch just outside, and then you're gonna head up the escalators to your tables where you have the chance to network with our current and past laureates. And thank you all for coming.